Yeah, for the lucky few decided to come today. We're gonna switch gears. So we've been talking about imaging stuff and optogenetic stuff in the previous couple weeks. But we're gonna talk about behavior for the next couple weeks. And we're honored that Matt, one of the world experts in this topic, is gonna give us a couple awesome lectures on this. I should also note for any neuro people in the audience, Matt's also an amazingly helpful consultant on any of your behavior analysis needs. So if you've been thinking about doing pose tracking or temporal segmentation or other behavioral neural analyses, we've got him for the next couple of weeks. So ask lots of questions.
Yeah, come talk to me. No problem. Yeah, so there aren't very many of you, so we can make this pretty informal. Please feel free to ask questions along the way. So like Liam said, we're going to talk about behavior for the next 2 weeks. So this week is going to be mostly about pose estimation. So going from raw videos to poses. And then next week, I'm going to talk more about things that you can actually do once you have poses. How do you analyze those? Okay, so I want to start off by talking about sort of the state of data collection and neuroscience experiments around 2018, because around this time was kind of an important sea change in the way people started recording behavior. So on the neural side, there were a lot of different technologies that allowed people to record data from the brain in different ways. So multi-electrode arrays had been around for a while. Neuropixels were starting to come online. There were a lot of different calcium imaging techniques out there, which I guess you guys have been talking about recently. There was also, tools out there for doing optogenetic manipulations. And for all of these different tools, there were a lot of signal processing tools that came along with them that were quite mature at the time. And of course, these have continued to be developed over the years and are getting better and better. But there were a lot of options that you could choose from when it came to analyzing this sort of data. On the behavior side, it wasn't quite so sophisticated at this time. So a lot of experiments, when they were looking at behavior, they were looking at these kind of very limited views of behavior. Maybe they were using touch or lick sensors or levers or treadmills or something like that. And so people were typically getting, sometimes just one-dimensional views of what was going on, the running speed or the pupil diameter or something like that. And it wasn't common at this time to use video data or other sort of high-dimensional ways of capturing the behavior. And so this was a ubiquitous enough problem that there's actually this nice opinion piece that was written in 2017 called Neuroscience Needs Behavior. And the argument of this article is basically that if we want to understand complex neural activity, we have to do it in the context of the behavior that the animal is performing. It's very hard to understand what's going on in the neurons themselves if there's no connection to the outside world. And so around 2018, there were two papers that you might have seen before, two very influential papers that came out. So the first one was a science paper from the Carandini-Harris lab. And basically what they showed was that neural activity during spontaneous behavior, it was really well predicted using videos of the face, of a mouse's face. And so this was kind of surprising, I think, to most people that this facial movement could actually describe neural activity in visual cortex, but also in many other parts of the brain as well. And sort of simultaneously with this, there was another paper from the Churchland lab that did a similar type of experiment, but instead of spontaneous behavior, the mouse was performing a decision-making task. And what they showed in this study was that neural activity was actually really well predicted by videos of the mouse's face and body, and not so well predicted by the variables that you might think are important, like the choice, or the decision of the mouse, the reward, and things like that. So a lot of the neural variability was described by the movement of the animal and not by these very specific trial events that we think are important. So There were labs that were using video to monitor the behavior of animals before these two papers. But after these two papers came out, I think they really convinced a lot of labs to start using video to record their animals during experiments. And actually, I think a lot of labs at this time probably recorded a lot of video data, not really sure what they were going to do with it. But this has become very standard now. And in the sort of 5 or 6 short years since then, a lot of really cool science has been done using video cameras as sort of the main behavior sensor. And so, as you might imagine, there are a lot of studies in motor control that are interested in tracking, in high resolution, what the different limbs are doing, rather than, for example, just using a joystick or a lever or something like that. There's a lot of social behavior experiments that are now sort of unlocked because we can actually track different animals and understand the relationship between these two animals at any given point in time. There's some really cool work on child development, trying to understand how little kids go from crawling to walking. There's cool work in animal conservation, tracking animals as they move by camera traps. And then, of course, something that is near and dear to my heart, some large-scale neurobehavioral correlation studies where we record a lot of neural data, record a lot of behavior data, and then try to understand the connection between those two. And so this is just sort of a sampling of the different subfields that have started using video data. And the question really is, how do you go from these big raw videos, which can be many gigabytes, and actually start to extract useful information from them? And so today, what I'm going to be talking about is one particular way of doing this, which is pose estimation. So pose estimation is not the only way to extract information from videos, but it's something that is very attractive to a lot of people. It's very interpretable, what you're getting out of these algorithms. A lot of people anyways just want to track, a couple key body parts that they're interested in. And so this is what we're going to talk about, is how to go from pixels to these poses. Okay, so we're mostly going to focus on 2D pose estimation today, but I also kind of wanted to situate this in the larger scheme of things. And so we'll start with centroid tracking. So for a time, this was kind of the only option that people had to do pose estimation in neuroscience experiments. And there were people that were doing centroid tracking mostly in flies, in dishes, going back maybe 15 years. And these were not using deep learning. These were using kind of older computer vision techniques like background subtraction and taking the median of these bright points or something like that. So they were relatively unsophisticated, but they worked well as long as the experimental setup was suited for this type of processing. So you can do a lot with centroid tracking. This is important for navigation and just kind of understanding where an animal is in an arena. But obviously, there's a lot that you miss out on if you want to understand more about the fine scale behaviors of the animal. And so here's an example of a mouse running on a treadmill. And obviously, centroid tracking here isn't going to give us a whole lot of useful information. We actually want to track these key points, track the paws, and try to analyze the kinematics of these paws. And so this is 2D pose estimation. It's going from videos to these points here. And so, like I said, this is mostly what we're going to focus on today. And then you can also do some really nice things if you have multiple cameras, and you can move to 3D pose estimation. So this is a little clip from the Aronov lab here at the Zuckerman Institute. And they actually have 6 cameras, and they're doing pose estimation in all 6 of these cameras. And then you can fuse all of that information together and get nice 3D trajectories of these different body parts. And so this is very similar to 2D pose estimation, but there's this extra step of combining information across views. Okay, so for the rest of this talk, we're just going to focus on 2D single animal pose estimation. I should also mention that there's multi-animal pose estimation. That is also kind of a step up in complexity, but I think you can understand a lot about that by really just understanding single animal 2D pose estimation. So that's what we'll do.
Any comments, questions, thoughts before we jump in?
Good. Okay. All right, so our goal here.
I mean, people are actually doing post tracking.
Oh, yeah.
We have people in the labs who are doing post tracking and. Thanks.
Nice. Yeah, so our goal here is going to be to automatically track a set of user-defined key points, hopefully using a small number of hand labels. So small number here means hopefully in the tens to hundreds. Maybe we don't have to go up to the thousands, but maybe we do. But this is something that we're going to talk about, is how we deal with the amount of data that we have to train these networks. So we're going to start with a set of frames, and typically someone will sit down and label the desired key points in each of these frames. And then the next step is basically to just train some sort of model that's going to map the frame into the key points that we've labeled by hand. So how does that work in practice? So typically the first step is some sort of feature extractor. So this can be like a convolutional neural network or a transformer, but some sort of neural network architecture where we put the image in, and what comes out is some sort of abstract representation of this image. It's going to be some set of feature maps, but yeah, it's going to be abstract and not necessarily something that you can easily interpret at this stage. But then there's going to be a second part of this model that's going to map from those abstract features up to a set of two-dimensional heat maps. And there's going to be one heat map per key point. And these are basically, you can think of them as like 2D probability distributions that tell you where the model thinks the key point is in the image. So for every key point that we're tracking, we're going to have one of these 2D heat maps. And then from those heat maps, we can extract the predicted key points. So for example, we can just have some sort of peak finding algorithm and say, this is the peak of this probability distribution. That's where we think the XY coordinate of this particular body part is. So we get the predicted key points from these heat maps. And then we basically just want to compare those to the labeled key points that we have from the data annotation step and compute some loss between these two things, figure out how close they are. Yeah. Yes, sorry, it is in the frame where the body part is.
Which could be totally different. Like it doesn't rely on the fact that the body part would typically be in the same areas, right?
No, so if you have a head fixed animal, for example, then the body part is probably going to be in the same area. In which case it's a much easier problem. But yes, this whole setup is agnostic to, what the animal is actually doing. And so it could be a large arena and the animal's moving around. And as the animal moves around, these heat maps are going to move around with it.
So there's like heat map for every frame.
Oh, yes. Sorry. Yes. So for every frame, there's one heat map per key point. And then if you put in another frame where the animal is in a different part of the arena, say, you're going to get a different set of heat maps. And so the goal of training this model is to train this feature extractor so that it can make that correspondence. So anytime you put in a new frame, it's going to give you a new set of heat maps that try to tell you where in the image that key point is. Okay, so this is sort of the rough overview of what this process looks like. Any other questions about this before I move on? Everyone's happy with that. Okay. So once you train these models, again, you push data through, compute the supervised loss, you change the weights in this feature extractor around so that these predicted key points look more like the labels, and then you just do this over and over and over during training until your model hopefully converges to some sort of suitable solution. The next step then is to take some sort of unlabeled video. You don't need to have hand labels for this. Push it through that feature extractor and also get the heat maps out. Take the peaks of all those heat maps, and then you'll get a time series of predicted key points. So on every frame, you'll get the XY coordinate for every single body part. And typically, the first time you train these things, the results are probably not going to be what you expect them to be. So there's typically we can't see it. There's typically a part where you go through and repeat this process. Go label some more frames, train your model again, put some more videos through, see how the results look, and then just do this over and over until you're satisfied with the output. Once you're happy with that, then you can go and do some downstream analysis. And again, next week we'll talk about some of these different downstream analysis techniques that you can do. Okay, so that's sort of a high level overview of what these pose estimation networks are doing. And so now I want to talk a little bit about the data side of things. So we talked about the models here. What about the data that we feed into these? So I want to start by saying that the use of pose estimation in neuroscience was really kicked off by progress in human pose estimation. So there was a lot of progress going on in human pose estimation in the early and mid 2010s, and that's what really convinced a lot of people on the animal behavior analysis side that this is something that we could do with animals too. The difficulty at the time, though, was that these early pose estimation data sets had a lot of data. So in this case, there were, this is one of the earliest data sets. There were 25,000 images. Some of them had multiple humans in them. So there were 40,000 total instances of humans in this data set. This particular data set was kind of humans in the wild. So you can see here, a lot of different backgrounds, people doing a lot of different things. There was also this human 3.6 million. So in this case, there were 3.6 million labeled frames, which is a huge amount, but these were this was a small number of people kind of in a more constrained setting and doing different activities with kind of like a black backdrop sort of thing. So this is a much more constrained data set, but there was a lot of data to play around with. So these are really big numbers. If you think about someone in a lab, some of you all have done pose estimation, so you've probably labeled some frames. Labeling even 25,000 images is a huge undertaking. And even if you wanted to, farm this out to some data annotators, this is going to take a while and be kind of expensive. And so people were kind of worried about translating this into the animal pose estimation world because we just don't have access to this sort of data. And at the time, the sort of wisdom of the day was that these neural networks would work really well if you had enough data. And so if you didn't have enough data, maybe they wouldn't work so well. So this was the question: is pose estimation even possible with a small number of labels? So this is where a couple sort of pioneering tools came into play here. So the first one I'm going to talk about is Deep Lab Cut, which I'm sure many of you are familiar with. And so Deep Lab Cut is solving this, what I'll call the labeled data problem, with transfer learning. And so the way that this works is they started with a model, a computer vision model that was trained on a really large image data set. So ImageNet, for those of you who don't know, is this big image database. And all of these images have some sort of category associated with them: truck, car, cat, hammer, whatever. And so this is a benchmark data set for fitting these image recognition models. And so what you can do is you can train a big model on this, say, take in the image, predict the class, take in another image, predict the class. So after that, you can actually take the weights that are learned from that model, and these weights have been trained on lots and lots and lots of data, but not pose estimation. And then you can build a pose estimation network. But the sort of backbone of this thing, the bulk of the weights in this network, are going to be the weights that we used in this first step. So we're sort of starting the network off in a good place. And then what we can do is fine tune it on a smaller data set. And the hope that is because the weights are already starting off in a good place, they kind of recognize different textures and edges and maybe even some animals. That's better than just starting this thing off randomly and hoping that it converges somehow. So this is what transfer learning is. You train a model on one task, and then you transfer the weights to another task, kind of initialize a new model, and then fine tune that model on a different task. And what they found was that this worked really well. And so this was one of the first demonstrations, at least in the pose estimation world, that this transfer learning idea worked. You could train these models with, you know, tens or even 100 images, and you could get some satisfactory results. Okay, so that was in 2018. So Deep Lab Cut has been out for 6 years now, and you know, it has close to 5,000 citations or something like that. It's been really, really popular, and a lot of people have used it over the years. And it's really enabled a lot of people to do a lot of cool science. So it turns out you can train these models on a small number of labeled frames. So that's not the only way to address this labeled data problem. Some of you might be familiar with SLEEP. This is another kind of big package in the animal pose estimation world. And this actually started as LEAP. The S is for social. So the first version of this was only on single animals. And their idea was, instead of taking these really big, say, ResNet architectures that were popular at the time, they proposed using a much smaller, lightweight architecture. So it's still using these ideas of convolutions, but there are just many, many fewer parameters. And the idea is that if there are many fewer parameters, then we need less data to train this thing. And indeed, that actually is the case. They were able to train these things with not a lot of data. So here is a sample efficiency curve. We'll see some of these later on as well. So this is looking at the error in pixels on the y-axis as a function of the number of labeled images that's used to train the model. So you can see even with 10 models, the pixel error is something like 6 pixels, which is not terrible. And this first image here is showing for each body part kind of, I forget what this is, something like 95% confidence interval, these larger orange circles. And so you can see some of these body parts are actually being learned really well. Some of the ones more on the periphery, the network is struggling with a bit more, but it's learning something here. And by the time you have, say, a couple hundred labeled frames, now you can see these 95% confidence intervals are much smaller. And so they're actually able to do a pretty good job of tracking in these fly frames with not a whole lot of labeled data. And again, it all has to do with the smaller architectures that they're using. They're not doing any of this transfer learning. They're not pre-training on anything. They're just training a small network on a little bit of data, and it works. Okay, so those are kind of two of the main popular packages that exist out there now. And so they each have their own ways of addressing this labeled data problem. And one thing that I want to mention is that both of these approaches use supervised learning. And so the idea with supervised learning, right, is we have this frame that we're inputting, and then we have the human labels that we're trying to match. And what I want to talk about next are some of the issues that arise with this. So, you know, both of these papers were very groundbreaking, and a lot of people have been able to use them, but they're not perfect. And so I want to explore a little more about the issues that crop up whenever you do this supervised learning with a small amount of labeled data. Okay, any questions on that before I move on? Yeah. Yeah, that's a good question. You could go look at, you know, the SLEEP paper, and I'm sure it would tell you that it requires less than DLC, and I'm sure DLC papers will tell you it requires less than SLEEP. Yeah, I don't think there's like a huge difference between the two. It's not like you would need 100 labeled frames for one and 10 times more in the other to get comparable performance. But that being said, I don't know of any studies that really thoroughly look at the performance between these two for a wide range of data sets. My guess is that it's probably pretty data set dependent as well. I wouldn't be surprised if one of these models does better on some data sets, but not others. Yeah, who knows? Also, you know, one thing that I'll say is, like, we'll come back to the sample efficiency plot later, but I think this is a little misleading because this is on held out test data, but it's test data from from animals that the model was trained on. So the model's already seen these particular animals. So if you show the model a different animal, then the picture is not going to look so rosy. These are also sort of simple because they've already been cut out of a larger video, and the flies are kind of centered and oriented in a particular direction, and they're very bright, and there's a dark background. So I think once you start to have more realistic videos, then yeah, this is not the type of curve that you're going to see. And so maybe in that case, something like Deep Lab Cut would do better because it has these pre-trained weights. But yeah, I don't know of any systematic study between the two of them. I'm sure lots of labs have done this like internally, but I don't know of anything published. I don't know if you know.
Empirically, it seems like DMC is a lot more popular than the
Yeah.
might be more popular than the version. See, but okay.
Yeah, I think there's also an important aspect of this too is the actual tooling that exists for these things. You know, it's one thing if one package performs much better than another, but if it's also much harder to use, then I think people are gonna say, well, I'd rather label twice as many frames than spend an extra 20 hours trying to install this thing. So that is important as well. Yeah, and I think that I mean both Sleep and Deep Lab Cut have spent a lot of time and effort into making their packages easy to run, but I'm sure people give up on one and go to the other because of some GPU driver issue or something like that all the time. Any other questions? Okay, so let's talk about the limitations of supervised pose estimation. So these are definitely important points to keep in mind if you're doing pose estimation right now, and also if you plan to do it in the future. These are sort of warnings for things to look out for. So the first one is that these algorithms are still pretty data hungry. So what I just showed you is that you can get reasonable tracking and you don't have to label 250,000 frames. But you might still have to label 5,000 frames or something like that. Of course, this depends on your experimental setup and the points that you're tracking and the species that you're tracking. But there is still a lot of data labeling that can happen in some of these projects. And this is an example from the Sawtell lab here at Zuckerman. And they have this cool setup where they have a single fish in a tank, but they have two separate mirrors so that you get 3 different views of the fish simultaneously. And they're tracking 45 body parts per frame. And so, as you might imagine, this is a very tedious set of frames to label. So this takes a long time to label one frame. And you know, they have 500 plus frames that they have labeled. And that takes time. And so it's something to keep in mind as you use these or think about using these algorithms, is that yeah, it's hard to say up front how many frames you need to label, but it might be more than you expect. So another point that's very important for actually using these algorithms for science comes back to that sample efficiency curve that I was talking about. So it's often the case that these algorithms fail to generalize to new labs or new animals or new camera positions. So to give you an example, this is a little clip from an International Brain Lab data set. So this is a head-fixed mouse. It's performing a visual decision-making task. It has this little wheel that it's using to indicate its decision. And here we're just tracking the 2 paws. We just want to know XY coordinates of the 2 paws. And so in this little experiment, we labeled 852 training frames from 5 different IBL labs. I should also mention, for those of you who don't know the International Brain Lab, it's this consortium of labs that are spread out across the US and Europe. And so there are around a dozen labs that are collecting data, and the data is supposed to be standardized and all look the same, but you know, it does look a little bit different from lab to lab. The mice look different from lab to lab. So this was trained using data from 5 IBL labs. Okay, so the performance is fine, but then we go to a test subject. So this is, you can see a very, very similar looking video, but it's from a lab that wasn't used in the training data set. And so you can see there's all sorts of weird things. There's this bias offset here. This marker is kind of noisy. It's switching back and forth between the two paws. And so this is, even though to our eye, these look very similar, these models can struggle with this sort of out of subject generalization. And so the way to really get around this is mostly just to label more data from a wider array of animals. Yeah.
So I guess I just have a question. In such a case, and let's say you have a lot of subjects, they might change, like I'm interested in development, so the mice size grows drastically. I guess my understanding was like you want to just make a general like training set and like have it generalized across, or do people tend to like train specific models and then like are okay with that like particular? I don't know if that makes sense.
Yeah, I mean, I think it's ideally you can have a single network that works for all of your use cases. Now, if the animals at the beginning and end of development look like really different from each other, then maybe you would need to have two different networks. But if it's mostly size and some sort of like morphological features that change a little bit, then maybe you can have one model for all of them. But you would need to train that model with a diversity of frames from different ages and probably different sexes and different rigs and things like that. So the advice that I would give is if you have some sort of labeling budget, say you want to label 1,000 frames, the worst thing that you can do is label 1,000 frames from one video. That's not gonna do well generalizing. And so the better thing is to take 20 to 50 labeled frames per video from a bunch of different animals. And maybe the camera can be shifted around a little bit. Maybe the lighting is a little bit different, but you really want to try to present your network with a wide array of different inputs so that it can kind of learn the data distribution pretty well. But you will probably find some sessions that it does really well on that it hasn't been trained on, and then other ones where it struggles a bit more. So that's usually this like repeat process where you say, okay, there's this particular type of animal or this particular situation where the model's not doing well. Let's label some more frames from those videos and then retrain and then try again. Okay, so yeah, so this is a really important thing, making sure that models generalize to new labs. So just to quantify this a little bit, I want to show you how important this can be. So this is taking the same IBL paw tracking data, and here what I'm plotting is the pixel error versus the number of training frames. So this is kind of like that sample efficiency curve that we saw from the leap slide. And you can see here that this plateaus around 100 or 200 frames. So this is similar to that plot in that regard. So adding more and more frames doesn't help us at all. However, if we move to those test sessions and animals and plot the pixel error on those, then you can see that there's a huge discrepancy between the performance here. And actually, even after 3,200 training frames, we still haven't closed that gap. This is a particularly heterogeneous data set. There's a lot of variability in the lighting and the camera position and stuff like that. But still, this is pretty. This was unexpected, I think, from me at least. And this is something that we were able to replicate in a lot of different data sets. It's not just this particular data set. So here's a pupil tracking data set, also from the IBL. You can see that also you continue to improve on these held out animals, even up to 1,600 frames. This is a social data set where it likewise takes about 3,200 frames for these the train and test animals to match. Here's that fish data set, and then that mouse data set that we looked at. So all of these are showing that there's really this big gap between the train and test sessions, and it takes a while to close that gap. Okay, so any questions on that before I move on to the third point? Yeah.
Do you have any insight into the network itself? Like that first example you showed where the on pause.
Mhm.
You had one that was offset, one marker offset, and one that was kind of bouncing back and forth. What's actually happening in the model that you trained on the first set of animals, to where it's picking up seemingly not features at all on the new animals, like our eyes can't even see like what it's looking at.
Yeah, that's a good question. The offset marker, I really don't know. I don't know what's going on there. With this one that's switching back and forth between the two, typically this happens when, so the network is trained to recognize the left paw and then separately recognize the right paw. But in this case, whenever you're asking it for where the left paw is, it's not really sure which one of these is the left paw. And so the probability distribution, that confidence map that it's putting over this image, there's usually like a bump here and a bump here, and they might be kind of equal. And that's what happens when this is kind of in the middle, and then it might kind of fluctuate back and forth between the two. So this is what we call paw switching, and this will actually come up in some future slides. But this can happen a lot, especially if you don't have enough data. The network, for whatever reason, just doesn't know which paw this is. It knows it's a paw, but it doesn't know if it's the right or the left paw. But yeah, I can't say about that one.
And it's not necessarily from overfitting on the first group of animals.
It's very possible. Yeah, it's possible that it's overfitting. And you know, there are ways of training these networks that try to keep them from overfitting. But you know, you can't really say for sure if you are or are not overfitting until you test them on some held out data. Yeah, so that's where like data augmentation comes in. You know, adding a bunch of augmentations to these images so that it looks a little bit different every time the network sees it. And then you can train these things with early stopping, so you can kind of monitor, you know, how well the network is doing on some held out data. And as soon as that starts to increase, then you can say, all right, we're probably overfitting. Let's stop there. But yeah, that's not going to totally get rid of the possibility of overfitting, especially when we're in these regimes where we're training quite large networks with not a lot of data. So that's always a possibility. Yeah, because this has 852 training frames, but this is using a ResNet 50, which has somewhere around like 20 million parameters. So yeah, it's a big network given this data size.
I think the left-hand side is more surprising for the right-hand side.
Yeah. Maybe you don't know, but do you have any idea how long it takes to actually, so if you have one frame, to make the estimations through like a network the size of DLC?
Yeah, so I would say DLC and sleep, the inference speed is highly dependent on the size of the image. But for the sizes of images that we deal with, you know, something up to 1080p or something like that, they can go maybe between 100 and 1,000 frames per second. Depends a lot on the GPU that you have and the network that you're using and stuff. But yeah, you could get easily 200-300 frames per second, faster than real time. Yeah.
On that sort of neurally relevant.
Yes. Yeah. And that's actually one of the selling points of the sleep models, because they're much smaller. The inference time on those are actually much faster typically, you know, given say the same hardware and things like that. So yeah, that's part of their selling point, is this can be very fast. Yeah.
So some of the videos you showed are pretty like smooth actions, so
Mhm.
they can move you seconds. But in our experiments, we're shocking the mice,
Okay.
and then sometimes so first I really thought that was just like the video was fast up, so the mouse would kind of jump around.
Oh, yeah.
Do you know, like we haven't seen a lot of problem going with like the DLC prediction, but do you know if that could be something that actually like we should look up to?
Well, so the frame rate of the video isn't really going to matter much for like DLC and sleep networks, because they're only processing one frame at a time. So they're not taking into account frames before or after that. So they're sort of agnostic completely to the frame rate. If you have a slow frame rate or you have like a really long exposure time, then you can have like a lot of motion blur or something like that. And that will lead to worse predictions because you just can't resolve the body part as much. But if like all of the frames are crisp and there's not a lot of motion blur, but you're only getting a frame every 100 milliseconds, let's say, yeah, that's not going to affect the models. I will talk about a model that we've worked on that does take into account the neighboring frames. And that's something that will probably work better if you have a higher frame rate. Yeah. Yeah.
A way to like take into account this the full skeleton or like how you know which nodes connect to which nodes that you can like you wouldn't have a pod next to it.
Yes, definitely. Yeah. So I will talk about exactly that in a few slides. So yeah, there are other tricks that you can use when training these networks to kind of keep them from doing the things that you don't want them to do. Yeah.
I have a question about how you treat the video temporal information during the training. Does the batch is randomly selected, shuffle or randomly shuffle the frames, or the frames are sequentially labeled or something?
Yeah, and pretty much in most of these networks, the frames are not sequentially, or they're not sequential. So you just have frame 131, frame 10,436 or whatever, and they just get shuffled randomly in the batches every time. Yeah. But like I said, we'll come back to the temporal part of this in a few slides. Cool. Okay, so now we'll move on to another problem, which is confident errors. And so the example that we'll come back to is this IBL paw tracking. So here are two example frames. And in this one, you can see both paws very clearly. So that's nice. The model can put these predictions on there. But then there are cases where one of the paws is hidden behind the wheel or up in the holder or something like that. And it's, the question is, there are several questions you could ask, but one, for example, is what does the network do with the marker for this paw that's missing? So I guess one thing that I failed to mention earlier was when we have these heat maps that are output by the network, you can also extract information about how peaked those heat maps are around their peak. So you can imagine if you have a really sharp peak and most of the probability mass of that distribution is in one point, then that's a very high likelihood prediction. The network is very certain that that's where the key point is. On the other hand, you could have that heat map that's kind of spread out in multiple places. Maybe it's putting some peaks on different body parts, or it's just kind of spread out because the model doesn't really know where that body part is. And so you can extract information, this likelihood metric from those heat maps by just saying how much of the total heat map area is concentrated around the peak of that. And so again, if you've used Deep Lab Cut or Sleep before, there's a likelihood column that you get back. So you get one value per body part per frame. And that's just the network's reporting, like how confident is it that that's where that body part is. And so here now I want to say that basically this can be wrong a lot of times. So here's an example where we're looking at the likelihood, this likelihood of the prediction. So it can go between 0 and 1. So 0 means the model is not confident at all in its prediction, and 1 means that it's very confident that that XY coordinate is where the body part is. And so what I'm showing you here are the likelihoods for a bunch of labeled frames where the paw is occluded. So the paw is actually missing, and the network shouldn't be guessing that it's somewhere with high confidence because we can't see it at all. And you see that there are a lot of frames where the network does output a low likelihood. So that's correctly signaling that like the model does not know where this body part is. But there are a lot of frames where the network thinks it knows where the body part is, and it's very confident about that prediction, even though this prediction must be wrong. So these are confident mistakes that the network makes, and these happen quite a lot. So again, later on, I'll show you some more examples of these. This is another way of looking at this problem a little bit. So this is now taking some labeled data. This is for key points that are visible. And we're computing the pixel error between the model's prediction and the ground truth label. And then we're plotting that as a function of likelihood. So what you can see is that there are a lot of points up here, a lot of key points, where the pixel error is very high, which means the model is very wrong, but the likelihood is at or very near 1. So again, this is a very confidently incorrect prediction. And there are some over here where the pixel error is very high and the confidence is low. So it's capturing some of these, but there's still a lot of mistakes that it makes. So this is something to be aware of as well. There's a lot of kind of downstream processing pipelines that people use that use this likelihood value and say, anything below some threshold, I'm going to say that's bad data, and I'm going to trust everything where the likelihood is greater than that. But that's not always going to be the case. Okay, last limitation that I'll talk about here is instability across seeds. So what do I mean by this? So for this example, we'll come back to this image I showed you earlier. So this is a mouse on a treadmill. We're tracking all of these key points, mostly just the paws. Those are the ones that are important for us now. And what I'm showing you here is the X coordinate, the Y coordinate, and the network confidence, this likelihood measure, for 1 second's worth of data. And so you can see this nice kind of rhythmic pattern as the mouse is running on the treadmill. And you can also see that this network confidence is at 1 a lot of times, and then it'll dip every now and then. And you can see here that there are some likely errors where this kind of rhythmic pattern is interrupted in a way that seems like it's maybe not real. And indeed, this is another version of that pause switching problem where a pause briefly occluded, this pause briefly occluded, and the prediction goes to another pause and then jumps back. And so some of these pause switches are correctly identified by the network confidence. So this one, for example, when this jumps up, the network confidence goes down. So that's great. But then there are other examples like over here where you can see what appears to be a big jump, but the network confidence really never goes too far away from 1. Now, the interesting thing is if we retrain a model and change shuffle the data up a little bit or initialize the model a little bit differently, then we can lay the predictions from a bunch of different models on top of each other. So here are the predictions from 5 different models. Each model is a different color. And you can see a lot of the times they agree with each other, but there are also times where they disagree with each other. And like over here, this is particularly messy. So this is interesting because on one hand, it would be nice if we could just train a model and then trust that that model is always going to give us the right output. But if we train another model slightly differently, then maybe it's going to do better, or maybe it's going to do worse. But we're actually going to later on use this feature of these models to our advantage a little bit. But I just want to highlight one of these frames so you can see what it looks like. So this is where the mouse has sort of slowed down and it starts to sit down a little bit. And so it mostly occludes the paw that we're trying to track here. And so these predictions kind of go all over the place. Some of them move to the tail. This one's just, sitting out kind of on the wheel. So this would be, one of these high disagreement frames. So we'll also come back to these high disagreement frames later because they're also the interesting frames. Okay, so that's everything that I wanted to say about the limitations. Are there any questions about that before we move on? All good. Yeah, so these are definitely things to keep in mind as you go forth on your pose estimation journeys. I would say just don't trust what's coming out of the networks because there's always going to be mistakes. So it's good to know what mistakes are being made in your data. Yeah.
A question or like a challenge I face is how do you know, when do you know it's good enough, right? Like where you like, can you trust them or not? I don't know if you're gonna talk.
No, I'm not really going to talk about it. It's a great question. And I think it depends to some extent on what you're trying to do with the poses downstream. So yeah, I mean, it's hard to give like one general answer that's going to work for everybody, but yeah, I think the main thing is just trying to do the analysis that you're doing and be aware of these different issues and see, I mean, if you have a way of figuring out how prevalent these issues are and maybe highlighting time points in your experiments where there's problems and then knowing to avoid those whenever you're doing analysis, then, you know, that's very helpful as well. And in a few slides, I'll talk about different ways of doing that, like identifying time points or frames where there might be issues. But that's a big thing, I think, is you're never going to be 100% perfect, but if you know when things aren't right, then that at least gets you something.
Yeah, I think that's a great question, and it's one we're still kind of struggling with on multiple projects. Some errors don't make any difference at all for the scientific question, and you can just happily run your analysis, and there's some corruption there, and it doesn't matter. That's fine. But some analyses are very, very simple for two things. Like if you really care about the velocity of this mouse's paw, then taking the derivative of one of these glitches is going to be fantastic. Right? So it totally depends on the scientific question. And I just wanted to echo what Matt said. You know, sometimes the hardest part of these problems, because you have millions of frames, you don't want to watch every second of every movie. So sometimes the hardest part is actually finding these problem areas and thinking about, you know, what metrics should I be computing? How should I be looking at the uncertainty? You know, to find those problem areas so I can go in and kind of quantify. So we'll that's definitely a big theme. We'll come back to that at the end. And I think there's still kind of open loop here. Are there other questions or comments about these points?
Okay.
You know, Matt has already mentioned a couple of times that it's a loop. And often you don't know at the beginning how well you need to track. You might not even know which body parts you care about, so you might have to go back and kind of add more body parts at the end. That's something we've talked about as well. Sometimes you're going to change your whole kind of setup, and you have to redo things. So there's, yeah, this scientific loop is something to see, and that's what people.
Cool. Okay, so now we'll transition a little bit to some of the work that we've been doing in this area. And so we were also interested in solving this labeled data problem and also just making these networks generally more reliable. And so we came up with this lightning pose project. And there were a couple key observations that kind of motivated our work in this area. So one of them was that we might only have a few hundred labeled frames, but we have lots of unlabeled data. Like Liam said, millions and millions of frames usually. And we're not doing anything with that data, or at least, you know, the standard supervised pose estimation models aren't doing anything with that data. So how can we leverage that data to get better pose estimates? And then kind of in parallel, we know a lot about moving bodies. They are constrained in time and space. So this, you know, is sort of what one of you was asking about earlier. We know that if you have you know, a wrist here and an elbow here and a shoulder here, you know, these two can't be here and the elbow be over by your foot or something like that. There are constraints on the way that bodies move. And so can we incorporate that somehow into these models? And so what we're going to do is talk about some architectures and some losses that take a few labeled frames, and that's the supervised part that we've been talking about. And then we add these mini unlabeled videos, and then we add to those unlabeled videos some spatiotemporal constraints. And this is kind of how we define some losses on these unlabeled videos, because we don't have labels, right? We don't know where the body parts are supposed to be, but we can say things like they should move smoothly in time, or body key points should change smoothly in time, things like that. So I'll talk more about those constraints on the next slide. So there are three pieces that we're going to talk about. So the first one is unsupervised losses on video data. So that's using all of that unlabeled data. The second one is incorporating temporal context frames. So some of you were asking about that. Instead of mapping from one frame to one set of labels, let's kind of look before and after that frame. And then the third one is kind of this nice post-processing step that we can do to clean up the remaining issues. So I might not get through all of this, but we'll start in on it and see how far we can get. Okay, so first I'll talk about these unsupervised losses on video data. And this was really spearheaded by Dan Biederman. He spearheaded this whole Lightning Pose project. And so what we wanted to, or again, coming back to this observation, we were thinking about poses and the set of spatiotemporal constraints that they should obey. What are some simple things that poses should do that don't require us to label data? So one of the things that we came up with is that these poses should evolve smoothly in time, and this is something that some other students in the lab looked at back in 2020, and then there's also been some other 3D pose estimation techniques that have used this idea as well. But it's pretty intuitive, right? Especially if you have a high enough frame rate, then these body parts shouldn't teleport somewhere to the other side of the frame and back. And so we can use that to identify problematic predictions. There's another really cool idea that we can use if we have multiple camera views. And so not every setup, of course, has multiple camera views, but when they do, we can use what we know about multi-view geometry to register if there's some error, if there's some lack of correspondence between the predictions on two or more different frames. And so I think, yes, so I'm going to talk in a little bit of detail about how this works, just so you can get a sense of kind of how we use these multiple views. So again, the idea is, let's imagine that we are recording an animal from two different cameras, and that animal, let's say we're tracking the nose, the tip of the nose of that animal. So that is a point that exists in three-dimensional space, right? There's an X, Y, and Z coordinate to where that nose is. When we capture it with one camera, we're transforming that, we're projecting that onto this 2D plane. So we get an X and a Y coordinate for the nose that exists in this plane. And then if we capture it from another camera, we're getting a separate X and a Y coordinate. This is a different projection, 2D projection of this 3D body part. So what we end up with is actually a four-dimensional description of this point, right? We have the X and the Y here, the X and the Y here. So more generally, we're going to have 2 times the number of cameras observations of this point that really exists in three-dimensional space. And so what we're going to do is we're going to, for all of our body parts and for all of our frames, well, again, we're just going to stick with this example of 2 cameras, but this generalizes to however many cameras we have. We're going to take this 4-dimensional vector, the X and the Y here, the X and the Y here. So that's going to be one of our observations. And now we have an observation for every key point on every frame. So we have this big matrix that is number of key points times number of frames by 4, because everything is a 4-dimensional observation. And now what we're going to do is something very simple. We're just going to fit PCA on this big matrix. And everything exists in 4-dimensional space. And we're going to say we want to keep the top 3 dimensions. And the hope is that those 3 dimensions explain all of the variance, right? Because we are just taking, because because this four-dimensional data here really is just describing something that lives in three-dimensional space. And so, right, the way this works is we just stack these up, x, y, x, y, project this down into a three-dimensional space that we learned through PCA. And then if we project that back up to 4 dimensions, the idea is that we haven't lost any information there because all of the information lives in this three-dimensional space anyways. So this is just an example from that mouse video that we saw earlier, because that's a single animal, but we see it from 2 different views. And here you can see the amount of variance explained as a function of the number of principal components that we're using. And so you can see by the time we use 3 principal components, we're explaining basically 100% of the variance in the data, which means that we are in fact able to project these 4-dimensional descriptions down into just a 3-dimensional space. And then if we add this 4th dimension, it doesn't give us any extra information. Okay, so okay. So the idea basically is whenever we're training our model, we're going to push this unlabeled data that we have through the model. We don't know where the body parts are supposed to be, again, because we don't have labels, but we know that the predictions from the different views should lie on this 3-dimensional subspace. And so we're going to project it down into that 3-dimensional subspace, project it back up, and hopefully there aren't going to be any errors between the two, between the original predictions and that reprojection. And if there are errors, it means that something is not correct in one of the views. We don't know what it is. We don't know which view is messed up, but something about it is not consistent between the 2 cameras. And so that error is something that we're going to try to minimize. And so we can do that through training, continue to push a lot of unlabeled data through the model and just continually check to see, does this data lie in a 3-dimensional subspace? Okay, so yeah, any questions on this? It's a little bit hand wavy, but hopefully it gives you a flavor of what we're doing here. Yeah.
So essentially you're using discrepancies in the X, Y components to estimate the Z dimension or the Z component, right?
Not necessarily. So it's more like ignore this for now. So imagine this is where the nose actually is. And if these are the 2 imaging planes of the cameras, then if we just draw a straight line between these 2, then this is where our predictions should be. So if the predictions are here in each frame, and we do this, stacking and then projecting down to 3 dimensions, projecting back up, the projections will end up basically right where they started. But if you have a prediction that is incorrect in one of the frames, then this configuration of one point being here and one point being here, this is no longer consistent with a point that exists here. And so if you do this projection, reprojection, then this is not going to end up where it started. It's going to move somewhere else. And so, again, you don't know where it's supposed to be, but you can say it wasn't supposed to be there.
But so when you have X1 and X2, does one of your principal components that you recover not predominantly align with some shared in X's?
I see.
And some in Y's?
I see. So that's going to depend on the data and where the imaging planes actually are. So that is kind of the case in the mouse on the treadmill example, because for example, in the bottom frame, the paws pretty much only go back and forth. Whereas in the top, they kind of go up and down and back and forth. But it's going to depend on the geometry of the cameras. If we were to shift the 2 views in a different way, then the actual principal components that we find would be different. So yeah, it really depends on the key point that you're tracking and how it's moving around in the different views and where the cameras are situated.
And then for this point that you're talking about tracking, maybe move into it, but how do you know it's the point you're tracking, without markers?
Um, so whenever we build the model, there's the feature extractor part. And then from there, we map those features to these different heat maps. And those are just, it's like arbitrarily ordered, but we say that like heat map 0 is always going to be the nose. And so whenever we're training the model, we say, you know, the ground truth XY coordinate of the nose should match heat map 0's prediction. And heat map 1 could be the top left paw or whatever. It's totally arbitrary, but as long as you're consistent when you're training and just say this heat map 0 is always where the nose should be. Then yeah.
Yeah, pulling out like arbitrary, maybe not arbitrary amount of features that are in the video. Right. So like when you're doing feature extraction, what kind of parameters are being set on that? I'm assuming it's like edges and
yeah. So we're just using these pre-trained ResNet 50s. So they've been pre-trained on ImageNet or some other big data set. And so, yeah, they are, you know, pulling out edges and corners and textures and things like that. The thing is, these laboratory videos look quite different than the images or videos that are used to train these big models. So we do find that fine-tuning the weights of the big model itself is also very helpful. So they do end up changing their representation through learning. And so, yeah, it's hard to say exactly what they're pulling out. I mean, it is going to be some, you know, combination of edges and textures and things like that, but that's going to be pretty data set specific after the training is done.
yeah, but the feature extraction is relatively robust across the different viewpoints of the cameras.
Oh, I see. You mean like if you train one model on multiple different camera views?
So how do you assure that the feature 0 in the left camera is feature 0 in the right camera for the 2 points of view?
Oh, I see. Yeah, that's just something that you have to set in a configuration file somewhere. So you as the user say, you know, I want to make sure that this feature in this or this key point in this camera matches this key point in this camera. So you just have to manually say like these are the correspondences, but you just set that once and then it, you know, during training that it takes that information into account. Yeah, good questions. Yeah.
This would be such errors and the key point is not visible to one camera, but it is to another, or like visible to not work.
Yeah, that's a good question. So if again, let's just stick with the 2 camera example. If a body part is not visible from one of the views, what the model is going to try to do is or let me say it this way, this error, this like multi-view PCA error is going to be minimized whenever the prediction lies on some line that's consistent with what you see in the other view. So it's not going to fully put it where it should be, but if you know where something is in one view, then it constrains it a little bit in the other view. And so it's going to move the prediction closer to that line. Once you have 3 cameras and you see something from 2 views, then it's going to be, you know, fully resolved. But yeah, this is very helpful with occlusions, especially if you have a lot of different cameras, because then you can say, I can't see this body part, but I know that it should be right there, given what I know from the other cameras. Okay, so the last sort of spatiotemporal constraint that I'll talk about is this low dimensionality of pose. So this kind of gets at the question earlier about skeletons and, you know, what is plausible and what is not plausible. And so to show you an example of what this looks like, so this is complementary to this multi-view consistency loss. This low dimensionality loss can be done with a single view. And the idea here is very similar, but instead of matching up key points across different views, what we're going to do is take all of the predictions from all of the key points, or a subset of the key points, and stack all of those together. So in this case, if we're tracking these 6 different points, we have the XY coordinate for each of these points, and we just put them in one vector that will be 12-dimensional, 6 key points times the X and the Y coordinates. So that's for one frame. That's the prediction from the model, say. And then on another frame, we're going to have a different pose. And we can, again, stack those up into a vector, and we can keep doing this for, say, every frame in a chunk of video or something like that. And now what we can do is play a similar game where we can say, let's project all of this pose information down to a lower dimensional subspace, and then project it back up and see how much information that we've lost. And this actually does work. So what I'm showing you here is the amount of explained variance in hand-labeled data sets for a bunch of different data sets. So forget about training a network for now. We're just taking poses that have been labeled by hand, and then just doing this sort of thing here where we project it down to 1 dimension or 2 dimensions or 3 dimensions and say how much of the data can we reconstruct with that number of dimensions. And so for all of these data sets, you can see that out here in the tail, there's a bunch of dimensions that are basically unused. So there's redundancy in these features. And again, that's because there are just certain configurations that are impossible, given where, say, the pelvis and the legs are, the neck can only be in so many different places. And so this limits the dimensionality of the poses that can actually be taken on. Yeah.
Sorry, just the valid poses, is that what it, does valid pose mean like a possible?
Yeah, so the way that we actually do this in practice is we compute this PCA subspace using the labeled data. So those are kind of like the valid poses. We say, we know that these have been labeled by hand. These are all real. Let's find the subspace that these poses exist in. And then let's make sure that when we're training, all of the unlabeled data that we're processing, those predictions lie in that same low dimensional subspace. And so, there are drawbacks to this, like there are ways that this can lead to some biases, but in general, it's a very good way of saying, like, is some key point way over there when the rest of them are over here, because that would definitely not be a valid pose.
I guess for this is really like a really wide variety.
Yes, and that's true of even just the supervised networks themselves. Just having a wide variety of poses is always good practice. And in fact, all of the different software packages out there, they have their own ways of kind of giving you a wide variety of poses to label if you ask them to. You can always say, I want to label these frames, but the default is to come up with a way of giving you a lot of different poses so that the network can kind of see all of the different possibilities. Yeah, so we did this little analysis here and found that there was a lot of there was low dimensionality to these poses, but we're not the only ones. Worms, people a long time ago, 15, 20 years ago, found that worms also have low dimensional structure in their bodies. There's a more recent paper showing this in human hands, and this makes sense, right? Like, hands are very complicated, but there are only so many things that a hand can do. So the set of possible hand poses also lives in a lower dimensional space. Okay, so that's kind of a tour through these different spatiotemporal constraints. These are just constraints that we played around with. This is by no means an exhaustive list. There's lots of other things that you could add to this. And so this is just kind of a starting point. But hopefully that gives you an idea of these types of constraints that we're talking about that we can apply to these unlabeled frames. Yeah. Yeah. So during training, you mean? When training the model? Yeah. So imagine we have like a chunk of video frames that's coming into the model during training. So maybe you have 20 frames or something like that. For each of those frames, we'll construct this vector of the predicted poses from the network. And then we can project that down to some PCA subspace that we found previously with the labeled data. And the way that we choose that dimensionality is by, before training, we do this sort of thing where we, see how much variance is explained by a different number of components. And we always choose the number of components that explains 99% of the variance. So we say whatever that number is, that's the subspace that we're going to project down onto. So, and then that subspace stays fixed throughout training. So we take that predicted pose, project it down to that low dimensional subspace, and then project it back up to the original pixel space that it came from. And then we just go key point by key point and say, what is the difference between the original prediction and this reprojected prediction? And if all of the pose lives in this low dimensional subspace, then there'll be no difference between those two. And so it'll be an error of 0. So that's called a reprojection error. So that reprojection error will be 0. If there's some mistake, for example, in the predictions, you project down and project back up, then there's probably going to be some discrepancy between the original and the reprojected poses. And so that's the thing that we're trying to minimize, is that reprojection error. Yeah. Okay. So wow, it went way slower than I thought. Okay, well, let me jump through. Let me not talk about this. The main point on this slide, this is just showing some another trace from this mouse on the treadmill data set. And basically the point here is that you can use these different spatiotemporal constraints to identify outliers. So we have the confidence or the likelihood, this is kind of the standard thing. But then you can see there are these like gigantic spikes that are probably errors, they definitely are errors. And the confidence doesn't register it so much. Or over here, this is a really bad one, it's barely picked up by the confidence. And so this is just showing what happens if you compute this temporal difference loss, and if you compute this multi-view PCA loss. And you can see that these get really big when there are apparent errors in the traces. And so the temporal difference loss and the multi-view PCA loss and the pose PCA loss, the one that I just talked about, they all kind of complement confidence as outlier detectors. And so they do a good job of highlighting frames where there's likely a problem. And so that kind of gives us confidence to say, okay, maybe these are good metrics to try to minimize during training, because they are highlighting outliers. And so that's exactly what we do. We take this kind of standard setup that we saw before. This is the supervised pose estimation setup. And now to use these unsupervised losses, we use the same feature extractor as before. But now whenever we're training this model, we take in a batch of labeled frames as before, but then we also take in a batch, a little clip of a video. And maybe this is 10 or 20 or 30 frames or something like that. And the labeled frames go through the model, and we get their predicted key points and compute that supervised loss as we did before. But then now the video goes through the same model, and we get that time series of predicted key points. And now these predictions, we can compute those unsupervised losses on those predictions. And again, the whole point there is that we don't know what the correct answer is, but we are trying to encourage the predictions to be smooth and consistent across views and lie in some low dimensional pose subspace. And so we apply those losses, and then, we apply those losses, and then, that information gets back propagated through the network from both of these branches. And like I said before, the unsupervised losses that I showed you are just the ones that we've experimented with, but there are plenty of other types of unsupervised losses that you could use in this setup to help kind of guide the network in a good direction. Okay, so I'll finish with this slide. So just to recap, the improvement here is that the model can be trained on a much larger corpus of data. So instead of 100 frames, maybe now we can train it on 100 labeled frames and 10,000 or 100,000 unlabeled frames. So the model just gets to see more data, which is always good. There are some limitations to this. So we still have occlusions potentially and ambiguities, which you might not be able to resolve by looking at a single frame. If you have multiple views, then you're in a better position. But if you just have a single view, then sometimes you just don't know where the body part is. It's hidden behind some experimental equipment or the animal itself. And then the other limitation, which we'll come back to next week, I guess, is that these constraints are enforced during training, which is nice. But then at prediction time, we have no way of enforcing the model to output smooth predictions or low dimensional predictions or anything like that. So there is still a possibility of these glitches happening. And so next time I'll talk about how we can change this setup a little bit or do some post-processing so that we can enforce this at the very end of things. Okay, so I'll stop there. Next time I'll show you some actual results and show you that this actually works, which is good. But I want to show it to you with some other things as well. So we'll stop here.
Yeah. Just to kind of get people thinking, maybe jump into the open problems.
Yes. Yeah. So yeah, so some open problems in pose estimation. Yeah, because I guess you all are working on class projects, so just things to keep in mind. So this calibration improvement, which I talked about earlier, the networks tend to be overconfident in their predictions. So how can we do better with that? That's definitely an open problem. I'm sure many of you are familiar with foundation models in text and vision and video and things like that. Are there ways that we can train models that will generalize, where we can train with a lot of data and they can generalize across labs or video types or species? There is work being done in this direction, but there's definitely a lot of room for improvement there. So, hopefully 5 years from now, nobody will have to label mouse data. We'll just have a model that works for mice. But we're not there yet. 2 years. An optimist. Yeah, some things that we're working on is going down the multi-animal pose estimation route with multiple cameras. So multi-animal, multi-camera. So there's a lot of work to be done there. Active learning. So, we talked about this kind of human in the loop thing where you label some frames, train some models, see how they do, maybe label some more frames. How do you choose the frames to label on the next round in order to kind of improve the model the most? There are also ways of generating weak labels. So instead of you going in and labeling very carefully every single body part that you want to track, maybe you can use some off-the-shelf model that is kind of good, that can give you some what we call weak labels, and we can use those to help train the model. So we tell the model, don't trust these totally, but these do contain some information that are useful to you. What are good ways of doing that? It's an open problem. And then of course there's cool multimodal models where here we're modeling behavior, these behavioral videos, but we can add to that text or neural data or acoustics or some other signals from these experiments that are being recorded simultaneously. And maybe we can extract interesting things from the behavior and these other data sources that we wouldn't be able to do with either one alone. So that's a little more broader than pose estimation, but definitely something cool to keep in mind. More generic unsupervised losses, like I was saying. Yeah, I guess we don't have to go through the details of that, but there are some other ideas there. Okay, so I'll stop there. Okay, so we got through only a part of what I wanted to last time. So we're going to maybe spend the first half of this class going through the rest of pose estimation. And then we'll switch gears a little bit after that and talk about some things that you can do with the outputs of pose estimation to do some further behavioral analyses. So I'll try to stay on time a little bit more, but if you have questions, please feel free to ask. I'm happy to stop. Okay, so just to remind you of what we talked about last time, we talked about pose estimation, kind of broadly, animal pose estimation in particular. And I'll remind you that the issue there that we typically deal with is the fact that we don't have a lot of labeled data. So this is unlike a lot of sort of machine learning, deep learning problems these days where there's a ton of labeled data and you can build these really big models. And that's not the case with animal pose estimation. And so there are several approaches that people have taken over the years to try to be able to train these models, somewhat sizable models, with maybe only tens or hundreds of labeled frames. And so I started talking about lightning pose, the approach that we've been working on. And so last time we got through this first piece of things, which was using unsupervised losses on video data. So that was a way for us to label a small number of frames and train the model on that, but then also exploit all of these big videos that we have that do not have hand labels, but nevertheless have a lot of information in them about what the animals are doing. So for those of you who missed that last time, again, like Liam said, we recorded all of this. And so today I'm going to move on and talk about a couple other pieces that we've worked on over the years to improve pose estimation beyond this semi-supervised learning. Okay, so the next thing that we're going to talk about is incorporating temporal context. Oops. There we go. Okay, so the next thing we're going to talk about is incorporating temporal context. And so this is a part of this project that was led by Cole, another postdoc in the lab. And the motivation for this, before we get into the details, which I'll kind of breeze through a little bit, is for any of you that have labeled poses before, you'll probably recognize pretty quickly that there are some frames where it's really difficult to know where the body part is that you want to label. And sometimes it's helpful to like look forward a couple frames or look backwards a couple frames. Maybe you see two paws that are right next to each other and it's hard to tell like which one's the right paw and the left paw. Sometimes body parts are occluded very briefly, and if you just scroll back or forward in the video a little bit, then you can make out exactly where that body part is. And so all of the models that we've talked about up until this point don't use any of that temporal information. They just look at one frame at a time and make the predictions about where the body parts are based on that one frame. And so we wanted to allow the models to see into the past and into the future just a little bit so that they can make better predictions. And so this is the model that Cole came up with. And so there's a couple pieces to this model. So imagine we have just a single frame for now. Let's forget about these frames here. We have a single frame. And what we talked about last time, the sort of standard single frame pose estimation, is to put this frame through some sort of feature extractor, convolutional neural network, or a transformer or something. You get a bunch of features that come out of this. And then you put those through some, what I'm going to call here a static head. So that basically turns these features into some sort of heat map or set of heat maps. So these are the 2D representations of where the network thinks the body parts are. Okay, so now what we're going to do is augment this a little bit. And we're going to allow the network to not only look at this, this particular frame, but the 2 frames before and the 2 frames after. And this 2 is just the number that we use. It could, of course, be 3 or more, but this kind of keeps everything computationally tractable. And so what we do is we push each of these frames through this feature extractor. Each of them, so we get a set of heat maps for each of these different time points. And then what we're going to do is put those into this bidirectional convolutional recurrent neural network. And so the details of this aren't important. The only important thing is that this model is able to look across all of these different heat maps over time and output a single heat map for the center frame. So this doesn't require labels for all 5 of these frames. It just requires labels for the center frame. But then the network is able to use at least these features from the surrounding frames to try to do a better job of localizing this particular set of body parts at time t. So again, I'm not going to go any deeper into the details beyond this, but what I'll say is that this can help resolve these brief occlusions or ambiguities between similar looking body parts. And so to give you an example of that, here are two frames from this mirror mouse video that we looked at last time. So this mouse is head fixed and running on a treadmill, and we see the side view and the bottom view. And so here's frame 38 from some video, and we're tracking the different paws and the different views. And so on frame 38, we're going to focus here on this particular paw. And I'm showing you the predictions from two different networks. One is the baseline supervised network, and then the pink one is this context model. So on this frame, this paw is easy to see. It's very visible to our eyes and to the network, and so both models more or less make the same prediction. But then when we go to frame 39, you can see that paw has moved forward a little bit, and now it's not even actually occluded, but it's just kind of the same color as the fur in this area, and so it becomes hard to distinguish it from the actual leg. And so this baseline model says, well, this doesn't look so much like a paw, but this really looks like a paw, and so it's actually going to move the prediction to another paw. And so these paw switches are very common, and again, I'm sure some of you have seen this sort of behavior before. But this context model is able to continue tracking this properly because it can see this frame, and it knows that, that this body part is probably moving slowly across here. And so it's able to use that information to better localize this key point. And of course, it has information about where that paw is in the future and another frame in the past. And so it doesn't make this sort of mistake of switching over. Okay, so that's all I'm going to say about that model, but it's very cool and is very helpful. So the improvement that we get is that this model can utilize this temporal context. And actually, the way that we've implemented this is that the inference speed, so when you want to go process a new video with this model, it's basically the exact same speed as the single frame architecture. That recurrent neural network thing that sits on top is pretty lightweight. So this can still we can still run inference on this thing at hundreds of frames per second. The only limitation is that training takes about 5 times longer, because now we're pushing 5 times as much data through the network. But that's generally fine. You know, we train these models. Ideally, we will train these many fewer times than we actually use them to run inference on the experiments. Okay, yeah.
Is there like a frame rate per second of each of these you're supposed to like backwards or forward?
Yeah, good question. So I think we haven't really systematically studied this, but I think that this model tends to work better at higher frame rates when the body parts do kind of move slowly. I think, you know, in the extreme, if you were looking at like, you know, a 1 hertz video where like the paws are just all over the place and there's no easy like temporal pattern there to learn, then that model probably isn't going to help so much. So I think this video here, this mouse on the treadmill, is something like 250 frames per second. It's very fast. But I think, you know, yeah, maybe 30 hertz or below is pushing it. I think it depends on you know, the animal as well and how quickly it's moving. But generally the faster the frame rate, the better this is going to do, I think. Any other questions on this? Okay, so the last piece that I'll talk about now is, oh, nope, that's right. I'm going to show you some results now. So last time we didn't talk about results at all. So I'll finally show you some results, but I wanted to get through the unsupervised losses and the context model before I got to this part. Okay, so we're going to keep looking at this mouse on a treadmill data set. And what I'm going to show you, how I'm going to display these results, is the pixel error on a completely held out set of animals. And these results are going to be from models that are only trained with 75 labeled frames. So it's not a lot of data. And we're just trying to see like how much of an improvement can we get with these different tricks in this kind of low label regime. So I'll start off with deep lab cut. So this is kind of our just fully supervised baseline. So it gets, you know, 15, the pixel error is around 15. This number doesn't matter so much. It's more the relative changes between these models. Okay, so now this next one is using this context model that we just talked about. And so we see this nice decrease. You know, it's not huge, but this is definitely an improvement. And again, it tends to be on these particular frames where the model is struggling. And we're going to talk a little bit more about that in the next slide. If we now look at the model that use these unsupervised losses, so we have labeled data and unlabeled data, you can see there's a pretty big improvement in the pixel error. So this is great. The model is able to learn much better from all of this unlabeled video data. And then finally, we can combine these two. Actually, we can use the unsupervised losses and use this temporal context for both the labeled frames and the unlabeled frames. And when we combine these things, we continue to get further improvements. Okay, so that's nice. This was kind of where we were doing a lot of our development of these ideas, and we were happy to see this. And then we said, great, what happens if we move to training the model with a bunch more frames? And so we did that. We trained with 631 labeled frames. So that was the full size of this data set that we were working with. And this is what we got. So basically everything was performing the same. So this was a little confusing to us for a while, actually, because when we actually looked at the outputs of these models on top of videos, it looked like our models, these lightning pose models, were doing much better than Deep Lab Cut. And it wasn't really clear why we weren't seeing that in these types of plots. So there's a couple of reasons for that. One is that whenever you look at these, this pixel error, this is on held out hand labeled frames. So it's actually a small number of frames. I think it's something like 250 frames here that we're computing these metrics on. Compare that to the, thousands or hundreds of thousands or millions of unlabeled video frames, and it's maybe clear why our eyes were picking up on differences in these big videos more than the labeled frames. The other thing that we eventually came to realize is that not all frames are created equal, and so let me show you what I mean by that. So last week we talked about the fact that if you train 5 networks using a slightly different set of training data, then you can get 5 different answers from the network sometimes. And so in this case, I'm showing you a frame here with the predictions from 5 different models overlaid on top of each other. And in this case, they all basically agree. And so in this case, this is a pretty easy frame. None of the models are really doing anything different than the other ones. If we look at another frame, now the predictions from the different models are all over the place. And so these are what we call more difficult or ambiguous key points because the models don't agree at all. And so this is actually a really good way for us to try to understand, is a particular key point on a particular frame easy or hard? And so if the ensemble standard deviation, or the standard deviation across all of these different models, is low, then that's probably an easy key point. And if it's high, then it's probably a hard key point, or it is a hard key point because the models aren't agreeing with each other. And so what we're going to do is actually plot the pixel error now as a function of this ensemble standard deviation for the different key points. And so that way we can sort of see on these hard frames, how do the models compare? On the easy frames, how do the models compare? And so the harder key points are going to be over here on this plot where the ensemble standard deviation is higher. And so first of all, I'll put up here Deep Lab Cut, the Deep Lab Cut predictions. And so the way that we're doing this is it's actually kind of like a cumulative distribution. So at the left end of the plot, we're looking at all of the key points, basically. And then as we go further to the right, we're looking at fewer and fewer key points, but they're getting harder and harder. And so out here, for example, we're looking at only 20% of the key points, the total key points, but these are kind of the 20% hardest key points. And as we go out here, we're going to eventually hit 10% and 5% of the key points. So as you can see, the pixel error does indeed go up as we move to the right on this plot. And so now I'll show you some other models. So this is our baseline model. I didn't show you this on the previous slide, but this is kind of our implementation of Deep Lab Cut. The difference between these doesn't really matter so much. Here's the context model. So here you can actually see that it's not improving a whole lot, but it's definitely improving more on these harder frames or harder key points than kind of on the overall set of them. But now if we look at the semi-supervised model, we can see sort of across the board, we're getting better predictions here. And then finally, we can combine these again. And so this is the same plot that I was showing you earlier with the 75 labeled frames. And what we were looking at before was kind of along this dotted line here, looking at these errors. And so now we're really interested in, what things look like out here. And so if we move to the model with 631 training frames, you can see that over here where we're looking at all of the data all at once, there's not a whole lot of difference between them. But as we move to harder and harder frames, these sort of fan out. And now we are improving as we add the context and the semi-supervised or the unsupervised losses. And then finally, the combination of them performs the best. So this was, it took us a while to get to these types of plots, but these are really what we wanted to look at. We want to see how are the models doing on these hard frames, because on the easy frames, the regular supervised models are doing fine. Okay, so I think that's all I wanted to say about that. Any questions on this part?
Just have a thought.
Yeah.
That thought, but like how much do you think like some of the errors could be coming from like human labeling, manual error, like the training frames that you're feeding into it is not actually high quality, so it would reach bottleneck that you just like can't get better.
Yeah, that's a good question. So we actually did experience this in one of our data sets, not in this one, but there was a data set that we were working with that was a very complex data set, and it was a very hard data set to label. And then we found out after working with it for probably a year or more that it was mislabeled, or parts of it were mislabeled. And after fixing those bad labels, we went back and retrained everything, and the results were much better. So it's, we didn't. Well, yes, okay, I guess it's happened at least twice. Yeah. So yes, the quality of the labels is very important. And the model is going to struggle to do better than the data that you give it. And so having high quality labels in the first place is very important for getting these things to work. So it's also something to keep in mind when you're actually labeling your frames, is coming up with key points you can label very consistently. You don't want to have a lot of noise in the human labels themselves, or the models are just going to inherit that noise.
I think Sherry's question is maybe, are you down at the floor now, or is there room to improve?
Yeah, I can't say for sure if there's room to improve. I think the best thing to do would be to have multiple people label a set of these frames, and then kind of see like what is the pixel error of the individual human labelers, and you probably can't get any better than that. But for any of these particular data sets, I don't know where that floor is. But there are papers out there that have done this sort of experiment, have 3, say, people label the same set of frames, and then you can kind of figure out like how well the models are doing in relation to that interlabeler variability, or even intralabeler variability. I myself would probably label frames differently if I labeled 100, then came back a year later and relabeled those same 100. But yeah, we haven't quantified that in this.
Here's a related question, like, do you think if you have more training data with probably more variable, manual labeling, is it a better case overall because it has just more frames and data to train on? That, for example, you have a smaller set of training data, but they are higher quality and more consistent with each other. Like, in other words, like, with having more training data sort of overcome, some human error by, just.
Yeah, I think it depends on the type of errors that are in the human labels. If it's like there's a little bit of inconsistency in where you're labeling a paw or something like that, then yeah, I think more labels is probably going to be fine. But if it's like you're switching the paw identities accidentally, then adding more of that data into the model is probably not going to be a good thing. But yeah, I think it's probably good to prioritize a smaller, high quality data set than a larger kind of messy data set, which is not to say that you can't pull information out of that. I think it just requires being a little bit more careful with how you actually train the model. And ideally you'd be able to quantify how bad potentially some of the labels are. So it's just a different set of problems that come up, I think, if you go that route. Yeah, good question though.
No verification question. So the upper three models are like completely supervised. And
Correct. Exactly. Yes. Yep. So it really just comes down to computing this. So we have the predictions from the different models, and we don't use the pixel error or the ground truth at all to define like easy or hard. We just say, given the predictions from different models, how variable are they? It could be based off of anything, like you could, for example, have a supervised model, a semi-supervised model, a context model. So that's 3 models right there. And you could compute the variance over those 3. It doesn't really matter so much what the models are, as long as they're giving different predictions to things. But yeah, I think we actually computed, for these plots, we computed the variance over all of these models, actually. So we said, actually for each of these models, we have like, I think 3 or 5 repeats. And so there's many models that are going into computing that variance to make this plot. But there's no like right way to do it. It's just whatever models you have, what's the variance across those models. Yeah, and this is also useful, I should say. One of the reasons that we wanted to do it this way is because this is now also something that you can compute on unlabeled videos. So you can take some really long video that you have, and if you have trained multiple models, then you can run inference with these multiple models and use this as a way to identify potentially problematic points. And what we found is that this is way better than using the confidence estimates output by any individual model, because those tend to be, as we talked about last week, those tend to be problematic sometimes. And so this ensemble standard deviation or ensemble variance is another way of getting at kind of the uncertainty for a particular key point. Yeah.
I think I missed it, but the unsupervised component of this is training like next frame prediction.
Yeah, so it's training. There's a few things that it's doing, but one of them, for example, is just saying that things should be temporally continuous in time. So if you have an unlabeled chunk of video and you have the prediction of each key point, there shouldn't be any like big jump somewhere else. So if there's a really large jump, then you penalize that. And there's some other ones based on like if you have multi-view data, you can use information from multiple views, you can use the pose of the animal. But yeah, we can talk about it more, but it's in the lecture from last week. But and I should also say there are lots of different ways to do it. So next frame prediction could be, another unsupervised loss that you could add in. It's more about the framework, I would say, than like the specific losses that we used. Yeah.
Just a quick question. I noticed all of the images that you've been dealing with so far have been like black and white.
Yes.
Grayscale. So I was kind of curious to know like how does color play a role, like in terms of different types of learning. Was just curious if it would complicate more.
Yeah, so actually all of the networks that we train and Deep Lab Cut and Sleep, like all of these work on RGB data. For us, everything is grayscale. And so what we do is we just take in the grayscale values and duplicate them across the 3 channels. But if you have color, then the models can use that if it's helpful. Yeah. So that's kind of built into the backbones already.
Train model of fully RGB color and you ask it to predict making things on black and white. Or the other way around, right? Does it damage the performance?
It might struggle to some degree, but yeah, I don't know. That would be interesting to try. Yeah, everything that we've dealt with so far has been grayscale, so we haven't actually tried that type of experiment. But it'd be interesting to see what it does. Okay, yeah, last one, and then I'll move on.
For the temporal context, you mentioned you're looking 2 frames ahead and 2 frames back.
Yeah.
Is that the parameter you can tune in the model, or is that set?
So at least the way that we implemented it, it's set to be 2, but in principle, it could be anything. It's mostly just a matter of, there's kind of a trade-off between like how much temporal context you have and how much like training overhead there is. Because now, at least with 2 ahead and 2 behind, it's 5 times as much data. So also the batches are a lot bigger, so training is slower. So you could add another one, you could go +3, -3, but now it's 7 times as much data. And so, yeah, we never really experimented too much with that. I think the further you go out, the better you would do in certain cases, but yeah, 2 has worked fairly well for us, yeah. Okay, so I am going to move on to the next part, but good questions. So the last thing I want to talk about pose estimation wise is kind of following up on some of the stuff that we've just been talking about, this clever model ensembling. And so this is again some work that was led by Cole in the lab. So we've talked about these ensembles as a way of identifying frames and key points where there are potentially problems in the network predictions. And so just to schematize that a little bit, imagine we have a test video and we have, say, M networks that we have trained. And again, these, the way that we do it is just by making the train and validation data shuffle different. So there are other ways that you can get different networks, but that's kind of an easy way to do it. So we have M different networks, and you can push this video through all of these different networks, and you're going to get predictions for each of your key points on each of these frames. And here what I'm plotting is, for example, the X coordinate of one of these key points for this video clip. And you can see the individual models in gray. And then I'm just computing the ensemble mean. So just taking the mean across these M models and plotting that in black. And so, as we said before, a lot of frames, these agree with each other really closely. So these are kind of easier frames. But then you see periods over here where there's more disagreement among the gray, the individual models. You can see that down here a little more clearly too. And so these ensemble means tend to be a better match to where the actual key point should be. And then here are the ensemble variances that we've computed as well. So now what we're going to do actually is something that's a little bit more clever than using the ensemble means or the ensemble medians. And the idea is to put this into a model that we call the ensemble Kalman smoother. And so I won't go into the details of this, but the idea is that we have some sort of model where we have a latent state that evolves smoothly in time. And that latent state gets projected onto these ensemble means. So the ensemble means are like the predictions, the observations that we're trying to model. And we can map from these latents to these observations using these some of these spatial constraints that we have, like in the multi-view case. So there's a lot to unpack there that I won't for now. All of that is to say we can sort of take these ideas about spatiotemporal constraints that we talked about earlier and kind of bake them into this post-processor. And so what this means in practice is that we have this model where when all of the models agree, then this whole EKS system is basically going to output that value that all of the models agree on. When the models disagree up here, then this is the model knows that all of these different predictions are do not agree with each other, and that's when this sort of dynamics model and the spatial constraints come into play more. And so we can kind of smooth through these periods where the individual predictions are maybe a little bit messy. And so yeah, I can go into this a little bit. So here is kind of an example of the performance on this. This is one coordinate of one key point, but this is just kind of showing you an example here. And I'm showing you a bunch of different post-processing methods that people have used in the literature. So an easy example is the median filter. And so this is like one of the defaults in Deep Lab Cut. And you just take a sliding window and say the actual value for the center of this window should be the median of all of the values inside of that window. And so this is great if you just have a single frame where the key point goes really weird and then comes back. But if an error persists for multiple frames, then that's not really going to get fixed by this sort of post-processing. And we found that to be true even with the ensemble mean, for example. So again, the gray traces here are the individual networks. The color is the post processor. So you can see here when there's a lot of networks that kind of are confused about what's going on here, the ensemble mean itself looks a little bit messy. But if we come down here to these EKS models, you can see that they nicely interpolate through this messy period because they're using this spatial smoothness constraint. Down here, we're also using some multi-view constraints. And so again, I know I didn't really go into the details of this too much, but the idea is basically that we can use these ideas from the unsupervised losses and kind of build them into a post processor as well. And if we quantify this in the same way, where we're looking at pixel error on some held out frames as a function of key point hardness, so to speak, we can see some really dramatic improvements. So the gray trace here is the raw DLC outputs. In this case, these are all DLC outputs. And then you can see these EKS variants are the pink and the purple, and they really pull down the pixel error for these hard frames where the models disagree with each other. And that's even true when we go up to 631 training frames. These EKS models continue to really help out a lot, even though we're training with a lot of data. Yeah. Yeah.
You see after that, does it mean like you don't specify the relationship between the cameras, but you let the model learn it itself?
Exactly. Yeah. So what we actually do is we use PCA to find. So we want to model this post processor runs on just a single body part at a time. And so if that body part is seen from two views, say, we have the X and the Y coordinate for each camera, for each view. So our observation is 4-dimensional, but our latent state is 3-dimensional. So it's learning some, it's not the true 3D coordinate of that body part, but it is, some 3D space where that body part lives. And then that gets mapped up to these 4 dimensions that we actually observe from the two cameras. Yes, you don't need like the exact relationship between the cameras. You don't need to do camera calibration or anything like that. It's just using the statistics of the data and saying that these 4-dimensional observations actually live in 3 dimensions. And occasionally they don't, maybe when there's an error, and that's when the post processor is going to come and clean things up.
I wonder in case that you have the true 3D coordinates that you know from camera calibration, and then you also compute that, and then that's a huge. Like, can you use the true coordinates to teach the network to have like incorporate?
Yeah, so I think the unsupervised loss that is like the multi-view PCA loss, I think as long as all of the videos use basically the same camera setup, then this PCA trick works really well. If there are like big shifts between the cameras and you can't really compute PCA kind of across all of the data at once, then I think the camera calibrations from individual sessions or individual videos could help there. But assuming that the cameras are fixed and not moving, then this should all work without any sort of camera calibration.
We might be able to improve it with camera calibrations. We haven't tried that yet.
Yeah.
Yeah. Maybe if you could go back to the EKS slide before with this model diagram. So right now we kind of assume that the latent state gets mapped into the camera linearity. That's kind of one of the assumptions of this latent variable model. Of course, that's not true. It's kind of fisheye curvature. But we could replace that linear, the green arrows kind of represent these linear models mapping the latent space into the observation space. We could replace that with a calibrated camera model instead. I think in some cases that would improve the results we just haven't.
And then when you do that, do you implement like a reference point that's easy to be seen like on both cameras, or is it like you just do the regular labeling of body parts and let it like go back and forth and refine like both models?
So you're talking about specifically the EKS part?
Yeah, because like the model is trying to learn to label the body parts.
Yeah.
And like they're also using body parts to infer the relationship between the cameras, right? If you don't
Yeah. Yeah, so actually what we do in practice is we use these ensemble variances to flag frames where the models disagree and say we're not going to try to compute these multi-view constraints, the multi-view PCA with those frames. So we actually go through and find the frames where we think there are problems and get rid of those, and then compute this PCA subspace. And then we go back and use that hopefully higher quality PCA subspace to process the whole video. But yeah, it's definitely sort of like some heuristics in there of how do we identify bad frames, you know, how many of them do we get rid of? You know, so it's certainly not perfect, but it is kind of a very simple way to do this without needing like the camera calibration or any other reference points. We can just use the predicted poses across this ensemble.
The other place where this model goes wrong is if one of the networks is wrong but confident, right? So if all of the ensemble members get it wrong in the same way, this model won't be able to fix that. So that's kind of a useful diagnostic. If you're training your own models, you can look for these confident kind of mismatches between cameras. And if you find a lot of those, it's like having the labels make the networks kind of better, because this post processor is not going to be able to.
All right, so I think, I won't spend much time on this, but I'll just say that this post processor works on a bunch of different data sets, single animal, multi-animal, single view, multi-view. So it's a pretty powerful tool. It does have its failure points, as Liam just pointed out, but in general, this has worked pretty well in our hands. Okay, so, oh yeah, one more thing, I guess. This is just a cool demonstration of this. So this is this publicly available data set of 2 interacting mice. And this is a particularly challenging data set just because there's a lot of complicated behaviors that happen in here. And so what we're plotting here in red are the ensemble members. So these are our lightning pose models, and we've used context and we've used our unsupervised losses, all of the fancy stuff. But you can see there are still frames where there's a lot of big mistakes that happen. And it tends to be when one mouse is occluding parts of another, or this wire is kind of moving across one of them. And so you'll see every now and then the red points will go kind of haywire, like right there. And so the green now overlaid is the output of this EKS algorithm. And so you can see it's really able to use the variance among these individual models and this temporal smoothness to do a better job, particularly on these hard frames. Cool. Yeah, so that's a nice video. All right, so that is all I wanted to say about that. So just to wrap this up, some improvements that we get from using this clever model ensembling is that we can take these kind of miscalibrated confidence values that we get from the individual networks and replace them with ensemble variance to quantify uncertainty. And that goes a long way for us. And then we can also enforce these different spatiotemporal constraints at prediction time, which we weren't doing in the individual networks. We were using these constraints for training, but not necessarily for prediction. And now we can use them for prediction. The only real limitations are the increased compute. Now you have to, push your data through 3 or 5 networks instead of 1. And then, as Liam mentioned, these errors can still exist when all the ensemble members are confidently wrong about something, or they all agree on an incorrect prediction. Okay, so that is everything that I wanted to say about pose estimation. And so really what we focused on last week and kind of what I wrapped up here was how do we handle small amounts of labeled data? And so Deep Lab Cut was all about transfer learning, taking some pre-trained backbone and then fine-tuning it on a small amount of labeled data. Leap and Sleep is all about network design, making these much smaller neural networks that we can just train from scratch. And then Lightning Pose is mostly about semi-supervised learning, so throwing in a bunch of unlabeled data and trying to use it in clever ways. And I should say that you can mix and match these things. In the Lightning Pose work, we also use transfer learning. We have pre-trained backbones that we're using. We could also use these unsupervised losses on using smaller networks. So there's room to play with these things a little bit, and that's still some research that we're looking into. But hopefully this gives you kind of an idea of the landscape of these different models and what they're doing. All right, so that's the end of pose estimation. I guess we talked about sort of some open problems in pose estimation last time. I won't go over that again, but again, the slides will be up and shared with you all. So before we move on to post pose estimation, are there any other final pose estimation questions? All good? Cool. All right, so now we're going to move on to, I'll talk about that some other time maybe. Okay, so now we're going to move on to stage 2 of some behavioral processing pipeline. Now you've got your poses, what can you do next? And what I'm going to talk about here is going from poses to actual behaviors or actions. But again, there are many other things that you can do with the outputs of these algorithms. Okay, so we have our pose estimator. So this was a slide that I showed you all last time. And we talked about downstream analyses that we want to do. And so today we're going to talk about supervised behavioral segmentation, and I'll talk about exactly what that is. We're also going to talk a little bit about unsupervised behavioral clustering. So these two approaches are doing similar things, but one supervised, one unsupervised. And then, of course, you can do neural encoding and decoding. You can connect the poses to simultaneously recorded neural activity. There's a lot of things to do there, but I'm not going to talk about any of that. And then, of course, there are other ways that you can try to analyze this data as well. But we're really just going to focus on the first two. Okay, so to start, we will go over supervised behavioral segmentation. And so our goal here is to detect discrete user-defined behaviors in a video. And so what that means is we have some video of an animal behaving, and we want to do some pose estimation first. And then we want to try to add on some labels here and say the animal is front grooming on this frame or these sets of frames. And then, oh, we're only front grooming here. Okay. It could be doing other things as well. So we want to apply these behavior labels to these sequences of frames and say, this is when the animal is grooming, this is when it's eating, this is when it's standing still, etc. And so how do we actually go about doing this? So we're going to start with our key points that come out of whatever algorithm that we've used. And this is sort of a general pipeline that I'm going to talk about here. Like before, we're going to put those through some feature extractor. So this could be a neural network of some kind, could also be other types of models, but mostly we're going to focus on neural networks. And that's going to give us some sort of behavioral embedding. So at each time point in this time series of predicted key points, we're going to get some point in n-dimensional space. Maybe this is a 16-dimensional vector, 32-dimensional vector, or something like that. And then from there, we're going to map that to some class probabilities for the different behavioral classes that we're interested in looking at. And then we're going to compare that to the hand label for that particular frame. And then this comparison, there are different ways to do this, but you could, for example, use cross entropy for these two different distributions. So your model is going to output a distribution, but then your hand label is just going to be this kind of one hot vector. Yeah.
Should we have like multiple class labels for a frame if an animal, for instance, is like rearing and like roaming at the same time?
Yes. So everything that I'm going to talk about here is kind of assumes that the animal is doing one thing on a frame. But yeah, you can definitely, another way to go about this is to just fit individual binary classifiers and say, on this frame, is the animal doing action A, yes or no? On this frame, is the animal doing action B, yes or no? And then you can kind of train all of those in parallel. Yeah, there are different ways to do it. If you want to allow for the possibility of multiple behaviors at once, then you can. The way that I'm showing it here is just one out of n behaviors, but they're very similar in the ways that you would actually go about training these. Okay, so again, this is kind of a general pipeline here, and I just want to show you what the output of this looks like. So this is for this fly data set. In this fly data set, we have a few different behaviors. The fly can either be still or it can be walking. It could be front grooming with its forelimbs or back grooming with its hind limbs. And so this is just a walking bout. So up top are the key points that we're tracking. So we're tracking 2 key points on each of the 3 limbs and then a couple key points on the back of the abdomen. And you can see there's this section here where you see this kind of rhythmic activity in the legs. And so this actually, well, what the model will do is output these probabilities, right? Like I said, the behavioral embedding gets mapped to these probability distributions. And so on each frame, there's this probability distribution. So here, there's a high probability of being still. And you can see very briefly, there's this back groom probability that comes up and then comes back down. And then the walking probability comes up and stays high for this entire segment. And so typically what we do is just set some threshold or just say, you know, what is the highest probability behavior at this time point? That's what's happening. If we want to assign an actual discrete label to this, but we can also work with these probabilities. That's totally fine too. So this is what a walking bout looks like. And then here are just some other examples of different behaviors. So front groom, for example, you can see that the forelimb bottom, forelimb top are moving and all of the other limbs are staying still. And so the model, as soon as that starts happening, the model starts to assign high probability to that front groom class and low probability to everything else. And then there are also times where, you know, the model can get kind of confused and maybe these probably, you know, no one class is really the clear winner. And those are actually labeled in white here. Those are kind of undefined. But that's just me saying if, you know, none of the probabilities are above 0.75, call that undefined. But that's really just a visualization thing, doesn't have to do with the model so much. Okay, so yeah, that's just a very high level overview of this type of problem. You know, a lot of the interesting questions are in what this feature extractor is, you know, different ways of training these models. So I'm not really going to go into any details with that, but there's, you know, interesting things to say about that as well. Mostly I just wanted to, in the short time we have to go over this, get across the general flavor of this type of problem. So it's generally key points in, and then probability distributions over discrete classes out. I should also mention that you don't have to use key points, right? You could train this sort of model and put the actual videos into this thing. That's totally possible. There are software packages out there that can do that. It's, you know, kind of a matter of preference which one you use. I think a lot of people like to do pose estimation anyways in their videos because there are lots of other things you can do with poses. So people have poses lying around. If you're using poses, it's much easier to train these models because, you know, for a given frame, you maybe have 16 pieces of information rather than an entire image. And so the data are just much smaller. It's faster to train these, faster to, you know, play around with different ideas. Nevertheless, you could put videos in. And there are certainly, you know, depending on the key points that you're tracking and the behavioral classes that you're interested in, there could be classes that you've labeled by hand that you just can't discern with the key points that you're tracking. And so, you know, there's also a question of, can the actual behavior classes be discerned by the key points? But yeah, I think most people use key points to do this sort of problem. Okay, so I just wanted to give you a really quick example of something that we've worked on using this fly data. So this was a project with the Axel lab. They got published a couple years ago. And in this fly data, as I said, we're looking at, you know, when they're running, when they're sitting still, when they're front and back grooming. They also do this abdominal bending type of behavior. So we labeled a bunch of frames, we trained these types of models, and here you can see just the amount of time spent in each of these behavioral states for a bunch of different flies. So, you know, most of the time they're kind of sitting still, but they do a lot of running as well, and they spend less time grooming. Well, some of them groom a lot. And what we wanted to do, what Evan, the postdoc leading this project, wanted to do was also simultaneously record neural activity with this behavior and try to understand connections between the two. And so these mice are actually being imaged with a 2-photon microscope. And we're getting a lot, the activity from a lot of different neurons simultaneously, in this case, about 1,500 over the course of this 10 or 15-minute experiment. And so the interesting thing is that if we plot this ethogram down below, so just at each time point, we're coloring the time point by the most likely behavioral state, you can see that these bright bands where the neurons are really active, first of all, they happen all across the brain. They're all aligned with each other, and they all tend to be aligned with when the animal is walking or running. And so you can even take some smooth version of this running signal, which is in black here, and then plot the mean fluorescence on top of that, and it's an incredibly good match. And so there's a lot of activity all over the brain that's connected to just the movement of the animal. And we can look at this in individual cells as well and see that most cells are either correlated or anti-correlated with this running signal. But you can go further than that. So what Evan did is actually fit a regression model trying to predict single cell activity from these behavioral class probabilities. And what he wanted to do was figure out, you know, are there certain neurons that respond to, well, how well do the neurons respond to running? But then are there some that respond to, say, front and back grooming, for example? That would be kind of hard to find if we didn't have these front and back grooming labels for us. So for the most part, a lot of cells were explained by the running, but you can see that there are some that there is some variance explained by grooming. And so here are a couple example cells. And so this one, you can see even for these very short bouts of grooming, there's a big increase in the fluorescence. And so the cool thing is that Evan was able to go through this fly data set and find these neurons that respond to grooming. And they also tend to be either very close to each other in the brain. So this is a view of the mouse brain, and the gray dots are all of the neurons imaged in this session. And you can see there are two neurons very close to each other, but then also there tended to be neurons kind of on the mirror of the other hemisphere. And so there was a lot of symmetry in the neural activity that he found. Yeah, so I just wanted to give this as kind of an example of some things that you could do. So you can go, you know, extract these behavioral states from video data, but then you can also go connect that to neural activity, say. Okay, I think I'm doing good on time. So that's all I'm going to say about supervised behavioral segmentation. So just to list some open problems, since I know you all have class projects that you want to look at. So similar to the pose, actually, a lot of these are going to look very similar to the pose estimation open problems. How can we decrease the number of labeled frames? For that fly data set, I think there were tens of thousands of frames that I labeled. And, you know, it would have been nice to do half that work. So how can we decrease the number of labeled frames and still get good predictions? So, you know, there are better backbone architectures that we can try out. Also, you know, what I showed you in that initial slide was taking the markers themselves and putting it into the model. Maybe there's a better way to change that behavioral representation, do some sort of computations on the markers themselves or the pose estimates themselves before feeding them into the model. And it turns out that that actually does matter quite a lot. Again, similar to pose estimation, I think I probably put this one up there. Active learning. So if you want to label some frames and then train a model and then use that model to go select some more frames to label, what's the best way to do that? So that's the process of active learning. As part of that is also outlier detection. So how do you say you make one of these models and you look at the outputs on a couple little video clips and you say, all right, I'm pretty happy with this. And then you go run inference on 20 hours worth of data. How can you be sure that your model is actually doing a good job on all 20 hours without watching 20 hours of fly videos? So it's a really hard problem, but it's one that you should all definitely keep in mind as you train these types of models and process a lot of data with them. They are certainly going to fail. They're not always going to be perfect. So how do you figure out when they fail? Okay, that's all I've got for that. Any questions on the supervised behavioral segmentation? No? Okay.
So, and some people have done this kind of behavioral segmentation. Supervised or unsupervised or anything. So we have more boost trackers, I think, before than segmentation.
You got to start somewhere. Cool. So now we'll talk about unsupervised behavioral segmentation. And I'm sure there are also some methods in this class that you all have heard of before. So like key point MoSeq is definitely one of these unsupervised behavioral segmentation methods. VAME, B-side, these are all kind of in this class of methods. And so with the unsupervised approach, the goal here is to detect these discrete behaviors like we were doing before, but now we want to do it without any sort of user labels. So instead of somebody coming in and saying, these are frames where the animal is doing this and this and this, we're just going to throw it all in an algorithm and say, show me the discrete behaviors that the animal is engaged in. And so this is more about finding these sort of statistical regularities in the data and having a model that can identify pieces of time and say the animal is doing something very similar here that it is here and here and here. So how does that actually work in practice? It turns out there's a lot of ways to approach this problem. With the supervised behavioral segmentation, again, you can play around with different backbones and stuff like that, but it's a supervised model. So there's the input coming in, and then there's the hand labels on the output, and that's kind of it. With these unsupervised methods, there's a lot of different ways to do it. And there's a lot of different assumptions that each of these, so these are, I think, probably 4 of the most popular ones. They all have very, very different assumptions and model structures. And so they're all going to pull out kind of different aspects of the data. And so what I'm going to do is just focus on one of these just to be a little concrete. But if you're interested, you should definitely check out the other ones because they're all interesting in their own way for sure. So I'm going to focus on VAME because I think it's the most similar to maybe some of the other things that we've talked about in the sense that it's mostly just a neural network doing some stuff, whereas the other ones are not actually. So this is an image from a figure from the VAME paper. So just to show you their setup, they're mostly working with mice that are walking around in an open field. So they're imaging them from below. They're doing pose estimation. And then the next step is they're doing this egocentric alignment. So they kind of, for every single frame, they align it so that the animal is facing to the right here. And so you'll sort of see the animal like twisting and turning, but it's always going to be facing in one direction. And that's because they don't necessarily care so much about the absolute position of the animal in this arena. They just care about the relative positions of the body parts. So once they do this egocentric alignment, then they're going to extract some time series. And so these are the pose estimates. And so at this stage, you know, this looks essentially the same as the supervised step, right? We could label some of these frames by hand, and then we could use this extracted time series to predict those hand labels. But we're going to do something different now in an unsupervised way. And so what they're doing is they're basically fitting an autoencoder to this data. So they're taking this time series of key points and they're putting it into this model. And what is this model doing? So this model, the exact details of this don't matter, but you put it into some sort of encoding model that is aware of time. And so it's sort of processing this chunk of data forwards and backwards. But at the end of that, it's outputting a single latent vector for this whole chunk of data. And that latent vector has a mean and a variance. And so we're going to sample, or it's outputting the mean and the variance, and we're going to sample from these. So we get some latent vector Z. And so this latent vector Z, again, this is maybe 20, 30 dimensional, something like that. And it's supposed to more or less represent this entire chunk of data. So what are we going to do with this Z? There's two things that the VAME model does. So first of all, it tries to reconstruct from Z this entire sequence that was input into the model. And because this is much lower dimensional, this isn't necessarily an easy task, right? Somehow Z, this low dimensional latent vector, has to include a lot of information about all of these time points. And so it's going to try to reconstruct all of these time points. So that's one piece of this. And then the other thing that it's going to do is try to predict basically the next step ahead. And so it's predicting kind of a shifted version of this time series chunk. And so you can train the model like this. So you have a bunch of these time series chunks, put them in the model, try to reduce the mean square error between the reconstruction and the prediction. And at the end of the day, you get a trained up model. And what we really care about are these latents Z. So this is now a representation of the data that is no longer in pose space or in pixel space. It's now this sort of abstract representation that hopefully contains information about not only the pose, but how the pose is changing over time. And so this is the first step, is just to train this model. So there's no notion of clustering or discrete behaviors or anything like that. We're just going to train the model, get these latent vectors Z. After the model is done training, then we can do some clustering in this latent space. So now you can take your entire video, project it down into this latent space Z, and then you can either do k-means clustering, or you can fit some sort of hidden Markov model or something like that. And that's where you're going to get these discrete states. So you're going to assign for every time point, in this case, just a single discrete state. Okay, so what does that look like? So here is a segment of data that got fed into the model. And again, for each frame, we have what they call a motif. So this is one of the discrete states from the HMM, or one of the clusters from k-means. And you can see that there's, again, this rhythmic activity as the mouse is walking around this arena. And the model actually picks up these different phases of walking. So we're cycling between basically the same three clusters. And this is all unsupervised up to this point, right? Nobody has gone through and said, this is, this particular phase of walking, and this is this particular phase of walking. This is just being pulled out of the data itself. The model is learning that these sets of time points here look similar to these sets of time points, which look similar to these sets of time points. And now the question is, what do you do with this? Because what you get out is, this is cluster number 39, and this is cluster number 1, and this is cluster number 13. So, I think the amount of work that you have to, or the amount of time that you have to spend with the data is kind of inverted between the supervised and unsupervised approaches. With supervised, you spend a lot of time up front labeling your frames. You look at a lot of data, label your frames, and then tell a model to go match that. In this case, you can dump a bunch of data into the model, without ever looking at it. But then now once you have the outputs of this model, you have to go back and say, well, what is the yellow cluster? What is the dark blue cluster? And so you end up looking at a lot of data. You look at videos where you see, this little clip of dark blue here, this little clip of dark blue here, and you look at enough of these and you can see, oh, this looks like, the mouse is rearing up or the mouse is turning to the left or something like that. And so what they do in this paper is they take this set of clusters and, how to choose the number of clusters is another question, but typically you can start with something somewhat large, like say maybe 50. And then you can do some sort of hierarchical clustering of those clusters. And so this is really just, trying to, this is some algorithm that assigns a similarity to these different clusters. And so you can group them in this sort of tree here. So you can say, oh, clusters 3 and 39 and 24, these all are similar to each other. And clusters 25 and 47 are all similar to each other. So you can build up a tree like this. And at this point, there's still, there doesn't need to be any sort of human intervention here. But what they did in this paper, and what's very common or sort of crucial when fitting these types of models, is then to go through and kind of put a human label on these. So someone came through and said, all of these clusters here, if I look at little snippets of videos, these all look like exploration, whatever that means. There's some operational definition they have of exploration. All of these clusters here look like the mouse just sitting still. All these clusters here look like maybe here looks like grooming. And so again, this is where there's a lot of time spent just looking at these videos and kind of assigning human labels to these things. So the next question is how can you actually use these? And one thing that you could do is, like I showed you with the fly data, you now say have 40 clusters, or you could use these sort of higher level, more abstract cluster identities and throw those into some model and say, let's, try to predict neural activity with these, or try to decode these from neural activity or something like that. Something that's really interesting that they did in this paper and also happens in, so a lot of these unsupervised clustering methods papers is they'll do an experiment like the following. So they have these mice walking around in the open field. And in this case, they have two groups of mice. They have these transgenic mice that are, I forget what they are in this case, maybe like it's some sort of Alzheimer's mouse. And then green is wild type mouse. So they have these two classes of mice, and they basically want to see what is the difference in behavior between these two groups. And so what they'll do is go through motif by motif or cluster by cluster, and they are basically normalizing the amount, or they're computing the amount of time that the animals spend in these different clusters. And the way that they've plotted it here is they're normalizing it so that the wild type mouse is always has, that's the sort of baseline, however much time the wild type mouse spends performing that behavior. And then they can say, relative to that, how much time did the transgenic animals spend in that particular behavioral cluster? And then they can do some statistics on this and find some of these behaviors that are either up or down regulated. And so you can see this behavior here, whatever this is, motif number 4, is one that is really highly upregulated in these transgenic mice. And so you can then go back and say, okay, let's look at a bunch of clips of behavior 4 and see exactly what that is. And then you can start to understand, what are specific types of behaviors that differ between these two different groups of animals. And so that's a common way to analyze this sort of data. And this is, of course, something that you could do without even giving names to all of these things. You could just sort of run this type of analysis and then say, okay, well, this is the interesting thing that popped out. Let's go look at that one thing. Let's go look at this cluster and this cluster rather than looking at all of them. But generally, at some point, you'll probably just have to look at all of them. Okay, yeah. In what sense? You could, yeah. If the labels that you had were like more fine-grained, then you could classify or group those somehow if you wanted to. I think this becomes very useful when you have a lot of behaviors that are all kind of similar. You know, maybe one of these is like grooming fast and one is grooming slow, or one is grooming sort of like the snout, whereas the other one's grooming like the head. And they look similar, but they're different enough so they get clustered together. So you could do it with hand labels. I think it would require doing a lot of hand labeling of a lot of different classes, but yeah, you could do this sort of thing.
I wanna do those unsupervised algorithms and people like start like more using it for example, like human patients, like to predict, for example, if people have incidents or not, like doing those facial recognition. That's something that people start doing on not
No, so actually, there was well, I'm sure there are multiple people doing this, but there's a poster at Cosine this year using VAME to study humans with OCD, I think. And so it was like a top-down view of humans in just some room in the hospital. And they kind of just let them walk around like the mice do for like 15 or 20 minutes and just let them do whatever they wanted to. So sometimes they'd go sit, sometimes they'd walk around. I think there was a table with some books on it or whatever. So they have this camera from the top. They do pose estimation on these people, and then they actually ran this VAME pipeline like this. And they had two groups. There were healthy individuals, and I think it was OCD. And one of the things that they were looking at, for example, is the transitions between different behaviors. So you can set up some sort of graph, you know, with all of these behaviors and say, what's the probability of transitioning from one behavior to another? And they found that with the OCD individuals, they would transition between behaviors much more frequently. So they were actually looking at not just the total amount of time spent in any given behavior, but how often and how they transition between behaviors. And so they were able to make some preliminary statements about, you know, how often they switched the different behaviors that they were engaged in. So that's one example that I know of. I think that's at UCSD, maybe somewhere in the LA area. But yeah, there might be other examples that I'm not aware of. Yeah, but for the most part, I think these models, they tend to get used in mice a lot. That's mostly where I've seen them, at least. Okay, so to wrap up, so yeah, so we got 15 minutes. So I also wanted to talk about some work that we've been doing in the lab. That's kind of looking at supervised. Oh yeah.
One's favorite question: choosing the number of clusters, and also, is this analysis kind of require each behavior to be put into one bin, or there are undefined bins?
Yes. So in terms of the number of behaviors, one of the nice things about this hierarchical representation is it kind of frees you a little bit from making, like, you know, a definitive statement like there are 30 clusters in this data. Instead, you can say we made 50 clusters, and, you know, clusters 20 and 21 are basically the same thing, but, you know, they are similar to cluster 39 or something like that. So this allows you to be a little looser with how you define the number of clusters. That being said, I know that the people that use these algorithms have their own sorts of heuristics for fitting the models with some number of clusters, and then trying to see if there are any unused clusters, or you know, seeing if there's, maybe if you have 10 individuals, maybe one cluster gets used by one individual for 16 frames or something like that. Then you probably don't need that cluster. And then so they have their procedures for, you know, fitting with a lot of clusters, deciding how many they can throw away, and then reclustering with a smaller number of clusters. But it's all very heuristic. It's just kind of, you know, whatever works for people in their particular setups. I don't think there are any hard and fast rules necessarily. In terms of if you can assign one or more or none, yeah, I think it really depends on how you do this step here. So a lot of these methods, a big part of it is getting to the latent representation. or some sort of representation, and then doing a separate clustering step. And so, you know, that depends on the clustering step that you use afterwards. So like I said, here you can use a hidden Markov model. You could also use k-means. You could use any other sort of clustering algorithm that works in, you know, in-dimensional space. And so that really comes down to the particular method that you use. There are some, for example, like key point MoSeek, or I guess MoSeek and key point MoSeek, where these are actually based on a hidden Markov model. So in that case, you get a probability distribution again over the discrete classes that you have. And so in that case, you are limited, at least in the sort of way that it's implemented, you are limited to, you know, a set of behaviors whose probabilities all have to sum to 1. There are probably ways to relax that assumption, but just using the software as is, you're kind of limited to that. But that gives you a probability over all the different classes rather than, you know, you are in this particular class, in this class only. Hopefully that answered that question. Yeah.
In terms of, you know, or it sounds like at least the way they're structured, there's like dimensionality reduction and then there's clustering. And I guess in terms of like comparing or like choosing one of these softwares to use, like, are there any, I don't know. Yeah, any recommendations when you guys?
Yeah, that's a great question. So I think that's the first thing that I put in the open problem slide. Yeah, I'm sure there are labs out there that have kind of internally done some benchmarking of these different algorithms. If you look at the papers, they're all going to compare to the other ones and say we did better. So, you know, I'm not sure if those are really fair comparisons, or they try to be fair, but, you know, we'll always find that theirs is better in some way. So yeah, I don't know. It's something that is probably hard to answer generally. I think it's really going to depend on the particular data set that you have, the particular behaviors that are being, you know, expressed by the animal in the data set. And so, no, I don't really have any good guidance or insight on that. Yeah, I mean, this is something that I'm very interested in doing too with a bunch of data sets, is just comparing a bunch of these. And I think the real answer is going to be that it's not that one is consistently better than the other. It's that they're all going to pull out different aspects of the data. And so depending on what you want to get out of it, maybe one of these methods is better than the others. But yeah, this is where I think this is a much harder problem space than the supervised learning, because there you know exactly what you're trying to get out of the model. And so you can just optimize that. But with these, it's much harder to define what success is. And I think success is going to be defined differently for any given experiment. That being said, a lot of these are used for, well, at least these three, MoSeek, BAME, and B-side. Their main use case is animal or mice in open field. And so there, maybe it's a little, you know, that's a very specific setting where maybe it's easier to compare them. But again, I'm not sure the papers are going to give you.
I think we can say empirically, we've tried these methods and stared at lots of videos. Matt has stared at lots of videos. Other people in the group have stared at videos. And in the end, it's been a little bit frustrating to kind of come to solid conclusions on some of these methods, because there are always these weird syllables where you're not really sure what's going on there, or you're not sure why this syllable is split from this other syllable. So that's going to close on some comparisons of unsupervised versus supervised methods that I think we're out of this frustration we had with some of these.
Yeah, thanks for the segue. Yeah, so we've been interested in this, and just to show you some kind of an overview of some things that we've been working on, we'll come back to this fly data set where we have these ground truth labels of what the fly is doing on a bunch of different frames. And what we can do is fit a supervised model like we've done before. And you can see that, you know, generally this matches up pretty well. Sometimes there's some misses here, but for the most part, it's doing a good job. we've also been working on some semi-supervised models. So this is kind of similar to what we've been doing with Lightning Pose, where we have some labeled frames and unlabeled frames. Ignore this for now. I'm not going to really go into details there. But then one thing that we were interested in was taking an unsupervised method and saying, how well do the clusters that come out of this unsupervised method line up with our hand labels? And so we used key point MoSeek. And so down here at the very bottom are the classes output by key point MoSeek. And so this ended up being like 30, 40 classes, something like that. And what we did was we took those classes and tried to align them, take each class and assign it to a hand label on the training data set. And so we can relabel all of these classes. And that's what this KPM aligned row is right here. And you can see it does a pretty good job, right? Like when the fly is walking, it's kind of mostly picking up walking. You know, these colors more or less line up. But there's also some places where it really misses things. And so to quantify this, we computed this F1 score. So this is just a score for classification. And 1 means, you know, everything is classified perfectly. And you can see here that this TCN model, this is the supervised model, gets pretty high F1s. And then if we look at key point MoSeek, this light gray, then these F1 scores are much lower overall. So if we're looking at these high level classes that we're interested in, like front groom, you know, key point MoSeek is able to find things that look like front groom, but it's not doing a great job of like really specifically highlighting the frames where the front grooming is happening. And not mixing other frames into that. And we quantified this in some other ways too with these cluster homogeneity scores. I won't go into this so much, but again, basically the clusters that come out of key point MoSeek, you know, tend to mix together a lot of these different classes that we've hand labeled. And we find this across a lot of data sets actually. So what we've been talking about is this head fixed fly. We also have this freely moving mouse, which is what key point MoSeek was more or less designed to work on. But again, the F1 scores for these different behavior classes that we have hand labels for is not as good as the supervised model. We have a head fixed mouse. Again, key point MoSeek doing something, but you know, not exactly matching the hand labels. And then we also have this human gaits database where there's a bunch of different activities happening with legs. And key point MoSeek is not working super well there in our hands. And so this is not to say that these unsupervised methods don't work or that they're not good. It's just that they are not necessarily pulling out the things that we as human labelers pull out of these videos. And so it's something to keep in mind when thinking about these different methods. You know, if you are working with a data set and you have a small number of classes that you know you really care about, and you really just want to be sure that you have a model that pulls those out of the videos, then supervised methods are definitely the way to go. You're never going to do better than supervised if you know what you want. And then, of course, you can play around with these unsupervised methods, but that's a bit more of a rabbit hole to go down. You know, getting them working and giving you reasonable output for your particular data set. Okay, yeah.
Do you think that like key point MoSeek performing worse is a function of it like over segmenting like the behavior or like differentiating things that either we don't care about or we don't really see?
Well, in this case, at least it's not because it's over segmenting, because if it was over segmenting, if it's like here's one type of front grooming and here's a subtly different type of front grooming, which actually exists, you know, like I'm the one that labeled these behaviors in the fly data, and there definitely is front grooming that looks like this, and there's front grooming that looks like this, and I labeled it all front grooming. So, you know, a model should be able to pick that up as 2 separate clusters in an unsupervised way. But the way that we do this assignment here is we're saying, look at all of the times that key point MoSeek has assigned, you know, cluster 39, and what hand label is is most overlapping with that. And so if key point mostly has a bunch of different front grooming clusters, they would all get mapped to front grooming. So that's fine. It's more that it's coming up with clusters that have frames that we call two different things. You know, there's some front grooming in there, but maybe there's some still in there or walking in there or something else. So it's like the clusters, the key point mostly clusters contain multiple classes that we labeled at least. Yeah.
Sort of related note, do you think it ever struggles with behaviors that contain like a composition of multiple movements? And
Yeah.
for example, if there's a similar shared motif between like front grooming and back grooming, like I don't know how flies move, but like if they do go out first, then that gets like split into both those categories? Or, like,
Yeah.
gets counted?
So yes, I think there are smarter ways that you can set up these models. So an even simpler issue that's kind of similar to what you're talking about is if an animal is doing something slow and then doing it in other frames fast, that's going to get split into two different clusters. Maybe you want those in the same cluster, maybe not. But if the animal is doing it like, if it has like two speeds, slow and fast, then fine, put it into two clusters. But if it's this sort of, you know, continuum of speeds that it grooms at, and you're just sort of arbitrarily chunking this up into, you know, grooming slow, grooming a little faster, grooming faster still, you know, that's when I think the models tend to do worse because it isn't really like a discrete state. And so I think your example is like a more complicated version of that, where maybe there are certain sort of composite behaviors where there are two things going on at once, and the model is just going to basically not find that like the common behavior in there. It's going to basically say this thing is one behavior, and even if it's mostly overlapping with this other behavior, that's another cluster. Again, you can change the model structures to take these sorts of behavioral structures into account, but that's not how they're set up now, and that would take, you know, developing a new model type. So yeah, definitely would be a little complicated, I think, to pull out good clean clusters in that instance. Any other questions on this? Okay, so last thing is just to talk about some open problems in unsupervised behavioral segmentation. You'll see that these are much broader open problems. So one that I mentioned is, how do these current approaches compare across species and behaviors? So a lot of these have been validated, at least by their developers, in mice in open fields. I think moving beyond mice in open fields is probably a very important thing to do. And it's not clear how well these work on insects or fish or something like that. How to extend these to greater than one animal. So this is actually a really interesting and hard problem. It's already a hard problem with one animal. You can imagine wanting to find sort of behavioral motifs of social interaction. But these don't naturally extend to two. None of these approaches naturally extend to more than one animal. You know, even two is very hard. So that's definitely an open problem, is how to make these things work and be robust with multiple animals. How to evaluate these models. Again, as I said before, this is mostly very qualitative right now. You just kind of look at these motif videos or syllable videos and kind of see, like, do these look consistent across animals? Do these look consistent within animals? You know, there's a little bit more you can do than that, but there's no agreed upon way for how to evaluate these things. And then, you know, it is interesting to think about how to integrate additional signals into these models. An animal might be doing something that looks very similar behaviorally, but the neural activity or some other additional signal could really distinguish that behavior. So, for example, you know, a very simple example is if you have a mouse that's sitting still, just kind of quietly, that could be very different than a mouse that's freezing because of some, you know, some shock that was recently delivered or something like that. So behaviorally, those might look very similar. Neurally, those could look very different, and maybe we want to cluster those separately. So anyways, because some of these models are based on neural networks, you know, it's can be easier to think of ways to integrate these different signals into these models and do some kind of multimodal segmentation. And oh, the last one, yeah. So this is also kind of in this vein of like combining supervised and unsupervised. Can you run something like key point MoSeq or VAME? And maybe it's going to output some clusters that are really good and some clusters that are kind of junk. Can you take the really good clusters and say, all right, I'm gonna label these as a particular behavior and then train a supervised model on those outputs? Is that a way to kind of get the best of both worlds? So, you know, that's one example of figuring out ways to go back and forth between these two, but I'm sure there are other ones as well. Okay, so that is everything and reasonably on time.